varieties, a lot of color diversity, a lot of genetic diversity. So we should be very grateful to this plant collecting team to going out and collecting these. They ended up with about 90 accessions of very diverse collards that got boarded onto the USDA. And actually just a few years ago, no, last year, sorry, we sent letters out to about 30 of these original seed keepers. We had their addresses, just asking them if they would be interested in doing an interview so that we could capture a little bit more of the collard history and a bit more of the story, because these geographers basically got a GPS location, they got the seeds, they got the name of the person that passed them to, and there's maybe a sentence or two about the actual culture or the cuisines or the history of these seeds. There's very little information that goes along with the seeds, because it was driven by a geneticist that was interested in the seeds as genetics, not the seeds as story or heritage or culture. And so to me, that was a loss, but it wasn't the focus of that project. And so we were trying to like backtrack that a little bit. But most of those letters came back, returned to sender. The person we were sending them to wasn't available anymore, presumed dead. We actually only got 3 responses out of about 30 letters. So very, very real and present genetic loss from this project. Not genetic loss, like human loss. The seeds themselves are saved. So that was all happening in the early 2000s. 90 varieties of seeds were saved. The thing that kind of strikes me about this whole journey is, one, all these varieties that were collected, there was no documented history. That you don't see them in catalogs. Most of them didn't even have names. The geographers themselves said, oh, you can name these seeds like that, because they were just like backyard collards. And so most of them named them either for the place that they were grown, for the person that gave them the seeds, or for themselves. So when you see all these seeds in the USDA, they're named for people or places. But there's no other documented history. So if this plant collecting trip hadn't have happened, then it's not even that we would have experienced the loss of those genetics. We wouldn't have even known that we missed the loss of those genetics. They literally would have just disappeared, and nobody would have known about them when those people died, because they were just, the genetics, the stories, the culture was only kept within those communities. So fast forward, those seeds got boarded into the USDA. We can like big check mark, that's great. The genetics were saved. But the thing that saddens me is that the people were still left behind. The communities were still experiencing aging seed keepers. There was still no system to preserve the genetics in place. So we see this kind of like weird system where we can go in and save the genetics, but the people are often forgotten. Their stories weren't even captured. And then what happens is those seeds that end up in the USDA become available to plant breeders. That's largely who gets to access those seeds. And then those seeds are grown out and developed into new and improved varieties, which are then often patented, but certainly some level of proprietary genetics comes into play. And then they're resold back to those communities as improved varieties with good marketing, which further undermines those systems in those communities to preserve their own genetics. And so we see this kind of downward spiral where the genetics that we want and we value as plant breeders are created within these peasant, indigenous, rural communities, but the product of those genetics undermine those communities. And so what happened about 15 years after these seeds were boarded into the USDA, Ira Wallace is a kind of a seed hero of mine. She is the matriarch of Southern Exposure Seed Exchange. She's done a lot of great work in the Southeast. And she happened upon a USDA trial of all these collards where she saw like these 60 varieties grown out. She's an aging seed keeper. She's in her 70s. And she saw all these collards and she was like, oh my God, this diversity of collards is so beautiful. Why don't we see this diversity in the marketplace? Why don't we see them? Why aren't the farmers growing these varieties? Why don't we see them at market? And so she kind of made it her mission to get these varieties out of the USDA and back to the communities and the people that are growing them. And that was kind of like the birth of the Heirloom Collard Project back in 2016. So it was almost trying to go full circle. These collards were in the communities. They were threatened because of aging seed keepers. They got boarded into the USDA, so they were saved, but then they were locked in the USDA or not accessed by the communities that best wanted them. And then Ira Wallace and the Heirloom Collar Project slowly been taking them out of the USDA, regenerating those seeds, and then getting them back out to the people. So I'm going to jump forward. Part of the Heirloom Collar Project is really about advocating for and promoting these varieties. And in 2020, we decided to do like a large variety trial just to try and get a little bit of energy behind these varieties, start seeing which varieties worked well where, and all that kind of stuff. And we worked with an organization called Seed Savers Exchange, Southern Exposure Seed Exchange, again with IRA. And we selected 20 varieties that we had enough seed of distributable quantities, and basically just invited the public to request to be a part of this trial. And we ran this through an organization called SeedLinked that did a fancy like triadic trialing system where folks would randomly be assigned 3 varieties of the 20. And then if we got enough people to randomly have 3 varieties and collect data on those 3 varieties, and it all got collected together. And we got some fancy charts saying which varieties did well where. So it was a nice little compact trial. It was a really good way to get community engagement with these varieties. We also had 8 farm sites across the country that grew all 20 varieties as a kind of like a demonstration plot. And the Utopian Seed Project was one of those trial sites. So we did just a simple 2 replication randomized block design on these 20 varieties. I had a 21st variety that I deeply, deeply love. So I kind of snuck another variety in. So I actually have 21 varieties in this trial in 2020. I'm not really here to talk about the trial too much, but that's what you're seeing here is this trial. We were doing the standard things. We were looking at yield. We were working with chefs to do taste tests. We were looking at plant structure, plant health, the standard variety trialing things. There was nothing too crazy about this. The collards were beautiful. They really, really, really were beautiful. One observation we had, which I think is really interesting and important, is these were technically heirloom collards because of their age, but within each variety, there was a lot of inherent diversity. So these weren't like hyper stabilized varieties. I don't know if they were diverse enough to be described as land races proper, but they were certainly not saved in a way that valued hyper distinct, uniform, stable traits. There was a lot of inherent diversity. And so within each variety, we might see some purple veining and some white veining that might different size plants, just small amounts of natural diversity within each population, which suggests to me that those community seed keepers that were originally keeping them, their purpose wasn't to have hyper uniformity. Again, their purpose was to feed themselves and their communities and save seeds so they could do it again next year. And that like strict value on stability and uniformity didn't seem to be there with any of these varieties. So we went through the trial site. It was a success, etc. We started going into that winter of 2020. And for me, the trial was kind of over at that point. Collards are obligate outcrossers. So having 21 varieties on top of each other, there was no way I was going to save pure seed off each of these varieties. And Seed Savers Exchange had a whole bunch of seeds. So seed saving wasn't really a component of this, even though in general, seed saving is a component of everything we do. So we kind of like, we didn't close it down because I didn't need that terrace. I just kind of let them go into the winter. We kept on eating collards and pretty much ignored them. And during that winter of 2020, 21, I forget the exact time, but we had one of those weird weather events that we're starting to get more often of late, where in the middle of winter, it'll be, in the 70s and the 80s for maybe like a week or two. Like we'll have, they call them Februnes, where we have these Junes in February. Just for a couple of weeks, like really hot weather, and everyone's like, what the hell's happening? Plants go crazy. And then at the end of that 2-week warm spell, when the plants were just waking up and wondering what was going on, we had this crazy Arctic plunge where it went from like 80s one day, and the next day we went all the way down to 8 Fahrenheit. So obviously, plants can usually experience extremes if they get there slowly in terms of highs and lows. But to have like a 70 Fahrenheit swing is pretty detrimental. And 8 Fahrenheit is pretty cold for collards, you know, at the best of times. So our collards were uncovered and we had this random event. And I live a couple of minutes from my farm, and I was just like, well, that's finally the end of the collards. I was just assuming that they'd all just be green mush. And so a couple of days after that weather event, I went to the field, this field where my daughter's standing here, and walked down the row. And it kind of seems like totally obvious in the retelling, but it shocked the hell out of me when I saw it. I walked down my row again, they're all in blocks, so like all different varieties. And there was like mushy plant, as I kind of expected, like just like no one's going to survive that type of weather event. But then next to that, there would be the beautiful green collared plant, as if it hadn't just experienced a 70 Fahrenheit temperature swing overnight. And it was just healthy and green, and I could harvest greens off it and eat it. And I was just like, what is going on? And that pattern was replicated across the whole field or row. So within each variety where there was that natural diversity, I was seeing some living plants and some totally dead plants. And there's no way that you could look at mushy plant and healthy plant of the same variety right next to each other in the same field, experiencing the same conditions, and not come to the instant conclusion that some genetic difference had allowed that plant to survive and the plants around it to go to complete mush. So the inherent genetic diversity within those plant varieties that had been stewarded by the community members had given them the tools to survive this temperature event. And that was a big light bulb moment for me. And so like there in the field, I decided just to kind of continue allowing that environmental pressure to select these plants for survivors. And whoever survived that winter without covering or any additional attention was going to be allowed to go to seed, and then we'd select the seed and plant them again as a big population. So pretty standard, although accidental, plant breeding project here, where we have this large composite cross, and then we're allowing the selection pressure of the environment to make selections for us. And so we posted about this on Instagram. You know, we're fairly active in our communications. And a friend of ours was like, oh, that's the ultra cross. And so this was the first example of an ultra cross that we created. And it was named by a friend on Instagram that was just like, you had all these different varieties, you let them all cross. It's called an ultra cross. And we selected for extreme cold survivability, beautiful diversity. And then because we work with a lot of chefs, then we're selecting for flavor. And let me jump forward and show you this. This concept has kind of become kind of like a bedrock practice of what we're doing with the Utopian Seed Project at the moment. At the time, I wasn't aware of this paper, but I've put it up there as a reference. If you've not come across it, I think it's extremely important, and it kind of talks a lot about what we do. I'll read the quote because I think it's powerful. Adaptation to climate change is a difficult breeding objective because of complexity, its unpredictability, and its location specificity. However, one strategy exists, which is based on a more dynamic use of agrobiodiversity in agriculture through the cultivation of evolutionary plant populations. So composite crosses, evolutionary plant populations, like these diverse, really broadly diverse intervariety crosses to create very diverse seed mixes. As a breeding strategy, they go into it deeply in this paper, but the argument is that in times of climate change, or more recently is being called climate chaos, where we have like this level of complexity that is unpredictable, and we don't know whether, like last year we had a really cold spring, summer, and then it went into a drought, and then it went into rains. But the year before that, it was early rains followed by drought. Like we're getting hail events, we're getting early frosts, we're getting late frosts. Every year is different, and farming is becoming more and more challenging. And if we're going down a path of traditional plant breeding, where we're trying to breed for a specific problem, and that problem is always shifting every single year, then we don't have the ability to use those tools in the same way to solve a problem. It's not as linear anymore. And so the argument goes that if we have a breeding goal that is highly erratic, climate change, or resilience to climate change, then we need to have like a highly adaptive breeding population to go against that. So these ultra cross or these composite crosses have like a really high adaptive capacity. And so by maintaining a high level of genetic diversity within the plant breeding population year on year on year, then the hope is that that plant breeding population has the capacity to withstand or survive or produce in conditions that the climate throws at it in any given year. So high adaptive capacity to meet high climate chaos is the underlying theory here. And again, this paper kind of lays it out beautifully and is well worth a read. So one thing we're doing with the ultracross, yes, environmental selection pressure is really important, and having these really diverse, tough, resilient crops, I think, is going to be critical and more critical in terms of having food security in a changing climate. But it also gives us the opportunity to select for kind of whatever we want. Like if we jump back to, that's the parent population there, which is fairly green leaves. Obviously, you're not seeing the plant structure or anything like that there. There's a little bit of purple coloration in some of these varieties. But very quickly, what we've seen is that the population is shifting and exploding into even more diversity, which is extremely exciting. And we've had a lot of kind of feedback from folks that we work with that they're very excited about the purple colors. The purple seem to be a little bit more pest resistant, probably because of the anthocyanins, but we've not researched that ourselves. But what we've started doing with these populations is huge genetic diversity and then selecting within that for certain agronomic or preferential traits. And this is an example of one kind of like line of the ultracross. We're maintaining like the hyper diverse population, but we're now splitting ones off. And this is the purple selection. And this is just pictures from a few days ago where, again, you can see this is just a couple of years into this, and we're already like heavily leaning towards these just beautiful purple colors. And there seems to be a high level of market demand for the purple line. So there's definitely the opportunity. This isn't like complete seed anarchy. There is the opportunity to make selections and have breeding goals within the diverse population. And to dig into that a little bit more, because I think it's important. We've still got the idea that, we still want farmers to be able to use these crops. And most of the farmers that I work with are small scale farmers and are not mechanized harvesting. So I appreciate that abandoning hyper uniformity is not going to lend itself to mechanized harvest. But that's not necessarily the agriculture that I personally believe in, so I don't care too much. And so with all the small farmers that I'm working with that are largely hand harvesting, then there's the opportunity for them to embrace diversity and have more on-farm resilience. And then it's a case of looking at this sweet spot. And I think this is something that I hold in the back of my head when I'm making these kind of crop selection decisions. What you're basically seeing here is a graph that demonstrates resiliency versus efficiency, and as it applies to diversity. So as we go towards monoculture, then we have systems that are like hyper uniform, and capitalism values efficiency above all else. And so that's why we see industrialized agriculture moving towards massive monoculture. The problem with hyper uniformity and a complete lack of diversity is that you create a system that is really, really fragile. And this is something that I hope everyone learned from the pandemic years, is that we have a system that is fragile. And when we have externalized shocks to that system, if there's no resiliency in that system, then it's prone to collapse or brittleness, as this graph says. So that could be like spiked fertilizer prices. It could be climate change and weather-related impacts. It could be supply chain shocks. But all those things in a system that has no resilience, because it's hyper efficient with no diversity, is prone to collapse. And I would, strongly suggest that we don't want to be living in a world where we have food systems collapse. In the other direction, if we go towards the seed anarchy, where we have like hyper diversity, then we have a system that is like insanely resilient. Like it could survive anything. If, as a farmer, I planted a field and every single plant was a different crop species, then I would be the most resilient farmer in the world. But I would have no efficiency at all. And how could I market all those different crops? How could I harvest them in any way that made sense? And my system would be prone to stagnation. It wouldn't actually work, even though it would be hyper resilient. So we're looking for this sweet spot. And this is a methodology in terms of making, having a breeding goal for these hyper diverse populations, these ultra-cross populations. I'm trying to find that sweet spot where I'm embracing as much diversity as possible, because I'm very concerned about resilient food systems, but I still want it to be an effective seed crop for the farmers that I work with. So just this is what kind of hangs out in the back of my brain as I'm navigating these crop selection choices. Actually, let me give you one example here. I'm going to come back to okra briefly, but okra, I'm sad to say, because I deeply love okra, our okra ultra-cross is an example of one of our failures. Like the collards are working beautifully, and farmers are growing them, and they're very effective. In 2021, I was excited enough about this that I set up an okra ultra-cross, or the beginnings of one. We planted out 100 different varieties of okra, and some of them were related species. I have a different breeding project that I'm not going to talk about right now, but it's more of a classic biparental cross to develop a high okra seed oil variety. So I had the 17 original parents in that population as well. All the parents of the okra oil breeding project are like from Sudan, and for some reason they're like hyper spiny and just like totally inedible, even though they have a high oil content. So anyway, I had this like hodgepodge of all these different okra varieties in the field. There's one related species called Abelmoschus tetraphilus that is just like this gnarly plant that produces totally inedible pods, but huge amounts of flowers. So like there's just lots of cool and interesting things, like okra geeky things going on here. And I decided at the end of the 2021 that I would just save seeds from all that, and that would be my ultra-cross. And what I basically ended up with is just like a mess. Like I am at the seed anarchy side of things, where when we grow them out, we have like 18 feet okra and 1 foot tall okra. We have like totally spiny pods, we have totally long pods and short, like we have everything in that mix. Like the diversity is astounding, and as an okra nerd, it's totally beautiful. But as something that I would ever suggest a farmer grow, like this is not it. And so what we've decided to do this year is keep that as a real, curious genetic pool, but we're going to restart the okra ultra-cross with much more intentionality about the parents that are going into it, and fall back to that 20 to 30 parent varieties of really outstanding varieties and allow them to cross, so that we can move forward with a better okra ultra-cross. So that, again, that's where that graph kind of like guides what we're up to, or in that case, it didn't guide what we're up to and it went wrong. Okay, so so far I've been talking about like what we've been up to. A big part of the work of the Utopian Seed Project and the work that we're doing is really trying to get it out to the communities and the farmers. And this quote has always resonated with me. With diversified crop species and varieties, farmers can adapt crops and their livelihoods to changing environments. So like, I think the power of this quote is that, again, kind of industrial agriculture relies on fairly narrow crop genetics to be spread over a large geographic area, and then asks the farmer to modify their environment to suit those narrow genetics. And that's where we see a high use of pesticides, fertilizers, all the other sides, fungicides, etc., as we're trying to like modify our environment wherever we are to suit these narrow crop genetics. But Dr. Carrie Fowler here is suggesting that we can, accept the environment that we have and allow diversified genetics to adapt to whatever that environment is. And so this sits very neatly in with this ultra-cross theory of regional adaptation based on diverse genetics. And I just got a very short example of kind of like that in action. It's very simple. Same collards. This is another one of those varieties, Tabitha Dykes, that we had a farmer growing out actually just outside of Durham. And she grew a row of these collards. Like I said earlier, there was natural diversity within each of these varieties. So she had a CSA customer box and she put collards in it. But that row of collards that she grew, most of them were green leaves and a few of them threw out purple leaves. And she got like instant feedback from her CSA customers that they loved the purple leaves. So she decided, based on that first year planting, to just overwinter the purple ones. Like she culled everything but the purple ones and just let the purple ones go to seed and saved her own seeds, planted them out the following year. When she had her row of collards for the CSA box, more of them were purple. She got more feedback from the customers saying, we love the purple ones. And just within 2 years, she managed to go from the green one on the left to the purple one on the right, just because there was inherent diversity within these varieties. And so this is just like a super simple example of using diversity and a little bit of farmer empowerment to like not just like localize, but like this is like she has done a very simple seed selection project to hyper hone in on a very specific customer need for her CSA box. And that's amazing. She didn't need to go through a whole breeding project. She didn't need to do anything. She just responded to change. And it was the baseline diversity that gave her that. And so she's able to basically be her own farmer breeder. I'm going to jump forward to this one. So based on that idea of a little bit of diversity, a little bit of farmer ingenuity, and community connection, then this is another kind of quote that has always impacted me. This is taken from Seedlinked. Seedlinked was that kind of crop trialing platform. Some of you may have come across it if you're doing any kind of participatory breeding projects. Then Seedlinked is becoming a pretty cool tool to be able to democratize data. And on their website, they have this quote where they say, increased availability and use of diverse seed would create more diversity in agricultural landscapes, contributing to breeding ecosystems that could evolve much quicker in the face of climate change, boost local adaptation and performance, and bring resiliency and food sovereignty to local economies in an era of globalizing fragility. So this breeding ecosystems concept is something that I think is just an amazing idea. And bringing it into the level of ecosystems and not just plant breeders and universities, but entire ecosystems where we have consumers and we have farmers and we have plant pathologists and all the other people. Like if it's a community effort and we're creating an ecosystem around plant breeding and not plant breeding as a separate thing, then I think we could see a lot of rapid and radical change within localized food systems just by empowering people to be their own plant breeders. And then if I jump back to this one, it's a different way, I feel, of saying the same thing. Robin Wall Kimmerer is just a fantastic thought leader. And she has written, using indigenous science, the human and the plant are linked as co-creators. Humans are midwives to this creation, not masters. The plant innovates and the people nurture and direct that creativity. They are joined in a covenant of reciprocity, of mutual flourishing. So the question for me is like, how do we facilitate those environments within the community where people feel empowered to engage with seeds and plants on that mutual level where they can co-create and be creative in creating more crop diversity? Because all of our crop diversity was created within the communities. And as plant breeders, we tap into that. But I think if we're not nourishing the plant communities and the people communities that continue to create that diversity, then basically we're viewing crop genetics as a finite resource, which is ridiculous because plants are always changing and always evolving and always adapting to change. So if we're not putting as much energy into the communities as we are into the science, then we're going to hit a brick wall. Okay, so I'm just going to finish this up just with a small example of how we're trying to do some of this work. And then I'll hopefully have about 5 minutes for questions at the end. So with creating these diverse breeding populations is relatively straightforward. Nothing kind of groundbreaking there. What we're trying to do is how do we use those diverse genetics to inspire or re-empower this community seed keeping concept that I want to see happen more often. And so one thing we've done with the Ultracross is just distribute them really, really widely. We've partnered with Southern Exposure Seed Exchange to sell packets of these seeds. So they're not distinct, uniform, stable varieties. They're not like in any way am I selling you something that you could even describe. I'm selling you a mix where like the one thing that is true is that every seed is probably genetically distinct. And every plant will look a little bit different from the one next to it. But we've had, hundreds and hundreds of people purchasing these seeds. This is just a map of the first year that we sold the seeds in 2021, sorry. There's been, multiple years of sales since then. So this will have expanded somewhat. Zooming in on the Southeast, those original college geographers, Dr. Ed Davis and John Morgan, they described Eastern North Carolina as the collared belt. Like that's where they found maximum collared diversity. So we're in a really rich area for collards, and then there's the whole cultural cuisine behind that. And so you see a lot of excited people about collards in this region. And then if we zoom in one more to the kind of the Asheville area or where we are right now, then we have more folks. Because this is technically a regional specific plant breeding project, then one thing we're trying to do is create kind of breeding swarms within certain geographic areas. So we have a Western North Carolina strain, but once the seeds go outside of Western North Carolina, I've had lots of folks reach out and say, hey, do you want me to send you seeds back? And they're in Southern Georgia. And I'm like, I don't want Southern Georgian seeds. Like that's your population now. We've just got someone's just grown 5 pounds of collared seeds up in the Pacific Northwest and of the Ultracross. And that's now, in my mind, a completely different plant breeding project. And so for us, the next step or where we're taking this next, this is still an active project, is we have all these dots on the map, all these people growing these plant populations. Some of them are not going to be saving the seeds, obviously, but some of them, we get feedback every year of them saving the seeds. They share the diversity. They're excited about this concept. And I'm working with a geographer from University of Kentucky at the moment, where we're just about to send out a follow-up survey to everyone that's received all those seeds to ask them, what are they growing? What are they selecting for? How are they interacting with these seeds, to try and start building a picture of whether have these diverse genetics like rekindled or restarted new plant stories within those communities. Because that's the baseline concept here, is there's a centralized plant breeding system where clever people breed plants and sell them, or there's a decentralized adaptive plant breeding ecosystem where we send out diverse genetics and have faith in both the seeds and the people to select in place so that they can continue to adapt and evolve and develop a deeper relationship over time. So I'll have to come back in a few years and tell you what the answers are, because we're still working on this and we're still encouraging this level of like community level plant breeding. I'm just going to whistle through a few of these. I've already talked about starting up the okra Ultracross again because of a small setback. We're working with southern peas. This is a mix we call the promiscuous pea mix. One problem with legumes is they're heavily selfing. So this concept of like high adaptive capacity and regional adaptation relies somewhat on outcrossing. So we've been selecting for outcrossing southern peas, which are these guys. We're working with a Machada and a Maxima mix. So lots of traditional southern crops. And And then just last year, we started digging into sorghum, and we were trying to work on a dual, like the agronomic qualities are to have high seed and high sugar production, but with a rainbow mix of diversity. Probably uniform height would be useful on that one too. Okay, I'm going to leave it there. I just have a couple of minutes for questions. But other than that, thank you very much. Yeah.
I think your dog is perfect. It's sort of a line in the size. I think you can really do something. I don't know if that's significant. Land streets, you see that we form. See like so I'm just saying that not all of them are responsible. On the different colors. Yeah. Did everyone hear that question? Nutritional value of the diversity. One very much interested in looking into that and knowing a little more about that. At our level of resources and technology, it's not something that we've been able to do. The one thing we do is kind of assume a loose correlation between flavor and nutrition, or loose nutritional density. And so we do taste tests. We work with a lot of chefs, and we do a lot of taste tests on our crops, and then make selections based on flavor. But we don't actually do nutritional analysis. But I would love that to happen. With a well-funded university. Well done. Yeah, I would love the university to be interested with that and to write a big grant and write into it and do the departments. Yeah.
Where does the Utopian Sea project sort of see itself in that relationship?
In the relationship between
the seed bank that is carrying the diverse sets of germplasm and participatory breeders who are actually growing the crops and making soil.
I'm with you. I think we see ourselves as liberators. We're trying to get seeds out of those gene banks and into the communities. And often there's either like an intimidation factor, a lack of knowledge factor, or often the USDA says no to people. And so we're lucky enough that they don't say no to us very often. So we're able to pull those genetics out, make some assessments, and then get them out to people that want to grow them. I'd say the other side of it is, as a nonprofit, we set ourselves up as a nonprofit because we knew we needed the flexibility and freedom to fail. Farmers don't have that freedom or flexibility, and often if you give them something and it doesn't work, then they're not going to give you a second chance. So as a nonprofit, we're able to kind of like shoulder some of that risk of this experimental side of things, get to a point where we're confident that it could be a benefit to a farmer, and then have that participatory side of things. When we have grant funding, we can pay farmers to be experimenters too. But I don't think it's fair to ask farmers to participate without compensation or inherent reward. Okay, thank you all.
And thank you, Chris.
Thank you, Chris. That was an amazing presentation. Next, I would like to introduce our next speaker, Dr. Massimo Lorizio. He's an associate professor here at NCSU. He's mainly located at Canapolis, NC, which is 2 hours from here. He is a geneticist working on small fruit and vegetable genetics and genomics. He works with a diverse range of crops. To name some, he works with blueberry, carrot, banana, pineapple, spinach, sweet potato, cranberry, and potatoes. He's doing some amazing work in small fruit breeding, mainly focusing on health promoting phytoactive compounds. I think that aligns perfectly with the theme of our symposium. So I'd like to invite Dr. Massimo for his presentation.
So, good morning, everybody. Thank you, Simon, in the introduction. Okay, please work. What do you want?
This is, yeah, I said I was sharing the wrong screen here. Is that right, Tiago? That's good.
Tiago, now. That's
Yeah.
working out, Tiago. So that was another recording.
Oh, that's your whole second. Yeah, that's the one it is.
But that is on the screen, so.
Yeah. So it's not sharing the presentation mode, it's sharing this screen. Is that visible, Tiago? Can the Zoom folks see that screen all right? Or is it too small?
No.
That's what we were doing.
What do they see on Zoom? Okay.
That's it. Okay, and the controls work? By using the arrow keys.
And the line, that's it.
You may have to click.
Okay. Okay, thank you so much. Sorry for the few minutes technical issue. So seems that everything is solved. So thank you again for the invitation. It's always the best pleasure when I receive invitation from a student for the symposium. So I guess the introduction just introduced who I am, and I'm faculty here at NC State with the horticultural department. Just a quick introduction for those that are not familiar with. I'm located 2 hours away from Raleigh at the North Carolina Research Campus in Canapolis. It's near Charlotte, 30 minutes out of Charlotte. And the campus there has been open now for since 2008, and the main focus of the research there is linking food and nutrition. And I'm located at the Plant for Human Health Institute. That it's this building here, which is the representation of NC State in our campus. In our campus, we have multiple universities from North Carolina represented there, and the Plant for Human Health Institute is representing the NC State. And then the mission in our institute is linking crops with health benefits. So this is my lab and the crops that I've been working on since I started here at Texas State. And you might recognize some of the students that are on my program. Now, my program, I'm a geneticist. I have a breeding training in my past, but being located in Kannapolis, managing a breeding program, it's challenging. It's relatively far away from one of the breeding stations. So my focus of my research is largely genetics, and then some pre-breeding, working collaboratively with a breeder to develop tools that can facilitate breeding for improved fruit and vegetable with an emphasis on quality. So in particular, what I look at, it's making a link between the DNA, genetics, fruit characteristics, and quality. Indeed, most of the time, fruit characteristics you breed, you can select for fruit characteristics, but you need first understand what is the connection with the fruit quality in order to understand what does mean quality. Okay. And that is some of the work that we do. It's to link the genetics with the characteristics, and then going to the quality, and then using this developing molecular tool that can use that eventually in the breeding program. So here, if you look at the breeding program, what is a common breeding program pipeline is. So here it's where you want to do your precision breeding and your tool. And my program focus largely into this area of establishing tools and knowledge for advancing molecular or accurate phenotyping, breeding with accurate phenotyping method in the realm of quality. So in my lab, most of the work we do, we develop genomes. So some of the genome we have developed for most of the crops, all the crops that I've been working on. And then nowadays, we passed the phase where we developed one genome. We are developing pangenome for the crop that we work on. We just completed a pangenome collaborative work for blueberry, and then now I'm working on a pangenome for carrot. So then how we use those genomic resources? So then we use for doing some structural analysis or understanding the genetics of the crop here in blueberry, where we did some comparative analysis to look at chromosome rearrangements or the genetics of blueberry being polyploid, auto or allopolyploid, which affect the inheritance of the trait. Then we give an emphasis on bioactives and understanding the biosynthesis of multiple bioactives, and then genetic study looking at trait of interest related to quality through QTL mapping or transcriptome analysis and validation of candidate genes. Among the other things, we use that information in some case to just answer some curious evolutionary questions. So here it's a fun project that I've been working on in the last 4 or 5 years now, and we recently published a paper on carrot, understanding orange carrot. We all know orange carrot today, but up to the 15th, 16th century, there was no orange carrot. Carrot was only purple, yellow, or white. And then we looked at the question, we used a large diversity panel, resequenced over 600 lines and asked the question, which are the genes that make carrot orange? And we found out that there are 3 genes. They are all recessive, and they make the combination of the 3 genes together. They are static. They interact with each other. You must have those 3 genes in order to have orange carrot. The fun part, they are recessive. All 3 genes are recessive, which make the phenotype of the orange carrot to be masked by other colors. So you can cross you can cross a white and yellow carrot, and out of it you can get an orange carrot, because those phenotypes are dominant over orange, so they are masked. In that sense, then we have been discovering genes involved in the understanding the carotenoid pathway. In carrot, we have been working on the anthocyanin pathway. We separate all the genes involved in accumulation of anthocyanin, which make carrot purple. In particular, we have been identifying a gene, which is an acetyltransferase that regulates the acylation of anthocyanin. And then we developed a molecular marker that can distinguish purple carrot that have high acylated anthocyanin and low acylated anthocyanin. Now, why is important the structure of the anthocyanin? The structure of anthocyanin in carrot is important because carrot in the last few years has been coming a common crop to use for natural colorant extraction, in particular the pink colors that you can obtain from purple carrot. And then we have been working with a collaborator from Argentina looking at the question, what does the structure of the anthocyanin affect the stability of the colorant use from carrot in different conditions? And it does. We found out that the acylated anthocyanin in carrot, when they use it as a natural colorant in yogurt product, are more stable at different pH and different temperature. So one of the main challenge for using the natural colorant extracted from our crop, it's that they are not very stable. So it's very challenging to replace the synthetic because of the chemical instability of the natural colorant. So acylated anthocyanin for carrot, it's are more stable and the molecular marker that I showed you, it's can shift from 20% of acylated anthocyanin to up to 100% of acylated anthocyanin. That means increasing the stability of those potential colorants. Another project that I'm working on is on blueberry, a crop that I focus a lot, and it's important crop for North Carolina. Here we are working on a large grant where we are looking at understanding the genetics of fruit characteristics related to fruit characteristics, and then how that link to fruit quality. Again, in a way to develop tools, phenotyping or molecular tools that can help to breed for improved quality. So all that, I think it's still on the realm of things that are relatively still common and been done in many labs. But for today's topic, I said, okay, why I don't focus much more the topic on the talk on trying to understand some work we have been doing on linking, trying to link DNA to health outcome of nutrient or bioactive that are derived from in our crops. This is a really challenging area to work on. It's not easy to establish those interdisciplinary research. And the reason why I chose this topic is because I think that it's important students realize that this is an area that could expand, and you will see how they can expand into breeding programs. So, as we know, fruit and vegetable are a source of nutrient, vitamins, bioactives, and then there are dietary guidelines that recommend certain amount of fruit and vegetable based on the content of these, based on basically on the fact that they contain nutrient and vitamins and minerals. And then there are a lot of studies that now show the health benefits associated with consumption of fruit and vegetable, with the consumption of certain fruit and vegetable that contain certain bioactives. There has been a huge body of study about the connection between some of those bioactives that I am listing here, flavonoid, anthocyanin, polyphenol, or carotenoid in the human health, this led to increase consumer awareness for healthy fruit and vegetable. And then nowadays also use of fruit and vegetable as a source of natural colorant has been growing. It's one of the market that it's growing the most in the food area. But when we talk about health property and nutritional value our crops, the way that they are evaluated by a food scientist or a nutritionist, it's by some parameters. So the first thing is what I call here the nutrigenomic property. By accessibility, which is the property of certain nutrient or bioactives to be released from the matrix, the food matrix. When you ingest a blueberry, the first thing that those bioactives like anthocyanin need to do, they need to be released from the cell and then be released and be exposed to the gut system. So that property is called bioaccessibility and can be measured. The second step is bioavailability. Once they are exposed to the gut system, they need to be absorbed in the circulation system. So that's called bioavailability. So it's the fraction of the metabolites that are in the circulation. Then the next property is the bioactivity. When you talk about anti-cancer activity or any other activity of a fruit or vegetable, it's because a bioactive or a nutrient has reached a certain target and then is having the effect that can have on the health. So when change the health status, that's a measurement of bioactivity. Now, all those properties, they are measurable. Food scientists, nutritionists, that's what they use for defining how nutritious or how good for your health are certain food, in particular here, fruit and vegetable. Now, there has been a lot of study to connect the nutritional property to external variable like a microbiome in human, the human metabolism, food in the interaction, food processing, and these come into the realm of precision nutrition. Today, there is also looking at the effect of nutrient of fruit and vegetable into epigenome and possible aging. But what has been not looking much is there an effect of the plant, the genotype, on the nutrigenomic property? All those studies are made assuming that every cultivar within a germplasm has the same nutrigenomic property for a given metabolite. Let's give an example, anthocyanin. The assumption is that any blueberry will have the same, any blueberry you eat will have the same health effect, will have the same anthocyanin being absorbed from any cultivar, and there is no study that asks the question, is that true? Is the time where we should start to ask the question and maybe find a way to select our crops for the real health benefits and develop method that we can select for health property, not only for what has been used so far by breeder, because in the field they need to select for something, and the most obvious one is most of the time it's either color, let's say a carrot that is darker orange, and in some case for content, if there are methods available for evaluating content. But when we talk about nutrigenomic property or other variable like the structure of the metabolites, there is no real much work done on the link between for selecting for a specific property. So to start to ask that question in 2016 now, it's 2017, with a group of colleagues from NC State, in particular from the food science back in Annapolis, we were awarded a project from DEFA Foundation, and we were able to ask that question. Is there an effect of the genotype on some of the nutrigenomic property? We focused on 3 crops, blueberry, spinach, and banana. And objective 1 was what I was involved mainly and was asking the plant genetics effect on nutrigenomic property. In particular, we looked at bioaccessibility and bioavailability of carotenoid in banana and spinach, and polyphenol or anthocyanin in blueberry. So the first step, and that's the big gap missing to do a breeding for and selection for nutrigenomic property, it's the phenotyping method. Most of the time, a food scientist will evaluate only 2-3 genotypes because there is no high throughput method for evaluating nutrigenomic property. So the first step was to develop a high throughput method for evaluating bioaccessibility. My collaborator, Dr. Ferruzzi from Food Scientists, he had already a small scale in vitro method for evaluating bioaccessibility for carotenoid. And then he worked on developing that method to scale up that method to a 96 well plate. And that was able, we were able to measure two property, relative bioaccessibility and absolute bioaccessibility. And the relative bioaccessibility measure the fraction of the metabolite that are released after digestion relative to the initial fraction that is in the matrix. And then the absolute, it's multiplied that by the total, okay? So then he developed the method, we validated the method with germoplasm that I was able to get available for him, and then the method worked, it was validated, so could be used for start to screen a larger number of plants for bioaccessibility using, of course, this, like I said, an in vitro method. The method can be used, and then we use this method for the 3-crop system that I showed. So what are the results, what we started to look at? So one of the systems we worked with is banana. Banana, it's one of the crop, one of the most eaten fruit worldwide. And it's also a crop that has been used as a target for biofortification, vitamin A, biofortification in many countries. So then it's very, very relevant, the work of understanding the carotenoid. Carotenoid are the precursor of the provitamin, the vitamin A. So understanding the effect of the genotype on the bioaccessibility of carotenoid for banana. There's been a lot of work done with banana in terms of increasing the carotenoid content. This is a paper came out a few years ago about developing a darker orange banana. And they increased a large amount of carotenoid in this banana. However, the literature of this group, the group working in the banana community, start to realize, this is a review paper, that there have been a lot of efforts on increasing carotenoid content in banana, but not much understanding, does this matter? Does it matter if you increase carotenoid in this banana? Is it bioaccessible? Is it bioavailable? So there has been no real study looking into that. So with my student, we looked at a question in collaboration with Dr. Ferruzzi. We selected a group of 30 bananas successions, and then we can see here the spread of alpha beta carotenoid. And then what we see is that there is a cultivar here, a local landrace that has relatively mid-level of alpha and beta carotenoid, but has 2-fold higher carotenoid bioaccessibility. This is the first step we did, the first case where we say, oh, there is an effect of the genotype, so content doesn't necessarily mean higher absorption of those metabolites. Then we did a similar study in spinach, where we looked at about 71 accessions for 2 years, and we asked exactly the same question. Does the genotype have an effect on the bioaccessibility of the carotenoid? And again, the answer was yes. There is a direct genotype effect that it's independent of the content of the carotenoid in these spinach lines. Okay, so the third model that we worked on was blueberry, and in blueberry we shifted gear. We worked on anthocyanin because in blueberry there is almost no significant level of carotenoid, rather anthocyanin or polyphenol are the most abundant bioactive in these fruits, and are the metabolites that have been associated with health effect on blueberry, and that has been the driver of increasing consumption for this crop. So using the same method, we started to look at the question, is there an effect of the genotype? So what we see, and then what is the relation with the other fruit quality characteristics that are used to select blueberry for? So fruit weight has no significant correlation with relative bioaccessibility. It means that the content, it doesn't affect the release of the metabolites, okay? Fruit weight has a significant effect on the absolute bioaccessibility. And the reason is because in blueberry, anthocyanin are accumulated only in the skin. So then when you have the same amount of fruit like here, represented, the same amount but different size, the surface area, it's obviously different. So then the small berry will have the larger surface area, the larger amount of anthocyanin. So that explains why absolute bioaccessibility in terms of total small berry will have higher absolute bioaccessibility. But again, fruit size doesn't affect the mechanism of releasing the metabolite from the matrix. So then we ask the question, does it matter the structure of the anthocyanin? Blueberry has a very complex chemical structure. Some anthocyanin, all anthocyanin are glycosylated with different type of sugar, arabinose, galactoside, and glucoside. Anthocyanin cannot be stable in water solution if they are not glycosylated. They are degraded right away. So they must be conjugated with sugar moiety. Okay, so then we ask the question, does the structure, the addition of the sugar, a different sugar, has an effect on the bioaccessibility? And the answer, it's not. So then after the glycosylation additional sugar moiety, there is another modification to the anthocyanin, which is the acylation. It's the addition of acyl group to the chemical structure. And then we ask the question, does the acylation has an effect on the release of these metabolites, on the bioaccessibility? And the answer is yes. So yeah, the acylation increase up to threefold the bioaccessibility of the anthocyanin. And the acylation is not something that you can distinguish by color looking at the berry. A breeder or a camera will not be able to select for high acylated anthocyanin because it's not visible. You cannot select for it unless you don't do HPLC. We'll ask the question, is the bioaccessibility of a bunch of metabolite, is it a heritable trait? Well, its heritability, it's relatively low to medium level. In some case, was relatively high, and of course, for which was the highest was for the acylated anthocyanin. And then we were able to identify lines that have high relative and high low absolute bioaccessibility. Now, the next question was, does this translate to human human system? This experiment are all done using in vitro model. The big question, does it hold true in human? So then what we did, we selected cultivars, some cultivars that have low relative and absolute bioaccessibility, and cultivars that have high, low, and high relative and absolute bioaccessibility. And then we worked, Colin Kay here from food scientist, he did a clinical trial where we used these two groups of cultivars to basically feed people for few months, asking the question, does this translate into a higher bioavailability of the anthocyanin? And the answer is yes. So the people that had eat the cultivar with the lower bioaccessibility, they end up being the one having the low bioavailability, lower absorption of those polyphenols versus the one that have been eating the cultivars with the higher bioaccessibility. So this, I think, it's one of the first example where we start to look at the connection between these structure of these metabolites and then the effect of the genotype on the bioaccessibility of the anthocyanin and direct translate to human trial. Now, how do we go by the genetics? So then we started to do, I'm just going to show you some example of the genetics on blueberry. So we did genetics of anthocyanin in blueberry. We know that these are highly heritable traits. And then we know that there are, just based on cluster analysis and heritability, we start to understand that genes with major effect probably regulate the addition of the sugar moiety, which is called the glycosylation, and then the addition of the acyl group to the structure, which is called acylation. Then we did QTL mapping for all the anthocyanin, and we found that majority of the phenotypic variation for accumulation of anthocyanin is explained by 4QTL, and those 4QTL don't explain much of the content of the anthocyanin, rather of the structure of the anthocyanin. So there are three major QTL that regulate the glycosylation, the addition of the sugar moiety, and there is one QTL in chromosome 2 that regulates the accumulation of the acylated anthocyanin. That is an enzymatic reaction, was relatively expected to find a major QTL. So then we started to look into candidate genes. For this, we did a transcriptome analysis and haplotype effect analysis, and then we could find candidate gene for all those QTLs. Here it's an example of the QTL for the glycosylation. This QTL basically affect the ratio between glucoside versus galactoside plus arabinocyte anthocyanin. Here you can see in the plot the recessive allele has very low glucoside anthocyanin, and this is this is percentage-wise, relative percentage of arabinocyte and galactoside. But then in the dominant allele, you see that the content of glucoside anthocyanin go up, and the fraction of the other two type of sugar moiety doesn't go drastically down, but still relatively high. And we found a candidate gene for this function, a UFGT, that we are now characterizing. Then this is a QTL in chromosome 8 that affect the ratio between galactoside and arabinocyte anthocyanin and overlap with a total soluble solid content QTL, and a sugar transporter was identified as the best candidate gene in this region. This is the acylation QTL in chromosome 2, and then this affected the fraction of anthocyanin, acylated anthocyanin. And in this region, we were able to go in the genome and start to look at the region spanning this QTL. So we found 5 acyltransferase belong to the BHDA family of acyltransferase. And then we did a RNA-seq analysis where we looked at the gene expression, and of those 5, only 2 are upregulated, 2 are not expressed, and there is 1 that it's not differentially expressed, it's expressed somehow, but not with a very specific pattern versus the line that have high acylated anthocyanin. So here we compared the line that have high acylated anthocyanin and low acylated anthocyanin. So that means these 2 are our best candidate for the acylation in blueberry. Now, why this is important? Going back to the link made with the bioaccessibility, the structure of the anthocyanin in terms of glucoside of the sugar moiety doesn't have much of an effect, but the acylation does. So then we started to focus more on the acylation, and then first step, we want to validate the QTL in another material. So a QTL is good for using for breeding if you can validate the QTL in other population in different environment. So that was our next step. So we did another GWAS diversity panel in another population grown in another environment, and then we found the QTL for acylation exactly on the same location. overlap with the one that we identified before. The top SNPs overlap with the acyltransferase. So the next step is we find those candidate genes. And, many times are just candidate genes. That's it. So until you don't prove the function of those genes. So then what we started to do is to validate the function of the genes. We did VIGs on a blueberry. We developed a new method for evaluating a function of the genes in berry fruit. And we silenced the 2 genes that we have candidates. We called VC81, VC82. This is the control. This is the line that were silenced, and then versus the control. And then what we see is that, first of all, the silencing of those 2 genes does not affect total anthocyanin at all. But when we look at that fraction of acylated anthocyanin, we go from a 30% of acylated anthocyanin to 0. So these are really the 2 genes that are involved in the acylation of the anthocyanin. And then next we ask the question: are we silencing by just a similarity? Are we using one construct that silence the other gene? Because these genes are highly similar between each other. The construct for VIGs could be aspecific and could be silencing the other gene. But then when we looked at the question, no, when we silence one gene in this line, for example, we silence the green in the transformed line, and then we looked at expression of the other gene is expressed. That means that the construct has not been silencing the first gene that it's in green. And then we did the same one on the other side. So we know that we have specifically silenced those 2 genes, and those are the 2 genes that are doing the function. So then that means that now we have 2 genes that we can target for starting to develop molecular marker and eventually start to breed and select for high acylated anthocyanin. Another project that I'm working on, and this is just really at the beginning, and just to understand the importance of looking into those metabolites. It's chlorogenic acid. Chlorogenic acid is another metabolite that in blueberry has been associated with health benefits. And there are multiple properties of chlorogenic acid. Now, if you look at the FDA on the website, they report blueberry to be one of the most important sources of chlorogenic acid. Now, that's true, but that's only true because there is a strong genotypic effect. Here you can see this is the table that the FDA has, but this is the distribution of chlorogenic acid in the blueberry germplasm. It ranges from 8 milligrams per watt for 100 grams of fresh weight to over 170. So the reason why blueberry is a major source of chlorogenic acid in the FDA table, it's just because there are some genotypes. There is a strong genotypic effect. So doing breeding for this trait that it's reported by the FDA, it's important with genetics because the only reason why it's in that table because there is a genetic effect. And of course, we identified the QTL for chlorogenic acid. We first identified in one population, and then we validated the QTL in another population across 6 years. And it's exactly on the same location, chromosome 2. And then the next step is to look for candidate genes. But another thing that I started to work with a collaborator from back in Canapolis from the food scientist, Dr. Andrew Nielsen, is to start to look at the effect of chlorogenic acid in regard to a metabolite. So, and here it's a short summary of it. So chlorogenic acid, one of the functions of chlorogenic acid is to affect the microbiota in our gut system, and then which produce this molecule. It's called the TMAO. The full name is here. It's a little bit complicated, but I like to call TMAO. So this TMAO has been associated with cardiovascular disease and risk of mortality in a lot of studies, okay? So chlorogenic acid seems to be modifying the microbiome in our gut system that produce way less of this TMAO, okay? So then what we did, I used my QTL data on the biparental population to identify lines that are high in chlorogenic acid, phenotypically, but also we looked at the QTL genotypic data. We selected plants that have the haplotype for a high chlorogenic acid, and they have indeed high chlorogenic acid, and the line that have low chlorogenic acid, and have indeed the haplotype block for low chlorogenic acid. And then what we are trying to do now, set up an experiment where we will use berries from those plants to then do an experiment using in vitro and animal model to ask the question, does high chlorogenic acid mean low TMAO? And that means a potential health effect. The preliminary data on in vitro already say that there is a dosage effect. So then that will mean if we are able to get funded, we have a proposal, another proposal for doing this research, it will mean that it will be worth for me to go behind and look for molecular marker for this trait. So again, does the plant has an effect on potential property, nutritional property of fruit and vegetable? The answer is yes. There is a study that came up last year. There are very few studies, very few work done in this area. There is a very nice study another study can use it as a model to support this concept. It's published in Nature Genetics last year, where they looked in sorghum. I didn't put up the paper. I forgot to put up the paper, but it's a paper in sorghum where they looked at GWAS in sorghum for changing the microbiome profile, and they were able to identify QTL that change the microbiome profile from shifting from a bad microbiome profile to a good microbiome profile. And what it's a tannin that is produced in sorghum that completely shift the microbiome. So, but the QTL was done for the microbiome, this property that the metabolite, the food matrix says. So again, we have multiple proof that the plants, the genotype has an effect on the nutritional property of our fruit and vegetable. The bioaccessibility study, it's one of the first that we have been doing, but also there is almost nothing in the literature that you can find that will trying to link genetics with a nutritional property. And this can help to expand and establish a new area of research to advance breeding for nutritional property. Now, where the big gaps are, the big gaps is the lack or limited interaction between plant geneticists, plant breeder, and food scientists. That's why I choose this topic, because I think that students need to be aware that there are opportunities in this area, there is work to be done, and if you like, there is a lot of opportunity. And then I call this my dream is the big QTL, or the big QTL. At some point, we will be able to do QTL implants for the bioactivity, bioaccessibility, or bioavailability. So, and like I said, this is we need more collaboration, and especially student cross-training between fruit science and plant genetics. It's very, very challenging to hire a PhD student that has some knowledge of food science that is interested on plant breeding, and vice versa. So with that, I want to acknowledge all my collaborators that have been involved. Of course, this is a lot of work, and then all the funding source that have been supporting some of the work that I presented here today. And then thank you for your attention, and remember, it all goes to the vegetable. Thank you.
Hey, thank you, Massimo, great talk. I had a cluster of questions surrounding the human clinical trials. I was wondering, looking at the graph very briefly, was it total anthocyanins, so therefore like high total anthocyanins and low total anthocyanins, or did you actually study the sugar moiety and the slate anthocyanins and human absorption?
Yeah, so what we did, we used, we selected based on the bio accessibility. I'm gonna go back to that. So here you can see in this plot, this is relative versus absorbed by accessibility. We selected the material based on the bioaccessibility of total polyphenol. The majority of the polyphenol were anthocyanin. So when we did the study, doing the trial take a long time to set up the RD for getting the permission to do the trial. So we had to select and also to find the material, because once you had to find the cultivars, then you need to find a grower. A research station doesn't produce enough fruit to feed people for a number of days. So it was a little bit challenging to set up the experiment, but to say that when we started to set up the experiment, unfortunately, we didn't know about the effect of the structure of anthocyanin. Yet, at that time, we were just analyzing HPLC, and we didn't know yet about the effect of the structure of the anthocyanin on bioaccessibility. So then we had to figure out a way to select the material. The best way was to select based on the total bioaccessibility, so low and high. So ideally would have been that we had already the data, and then we would select based on bioaccessibility and acylated and unacylated anthocyanin. So thank you for the question, because I want to just add, would I now recommend a breeder to select for high acylated anthocyanin? I would say not yet. Although there is no negative effect on selecting for it, there is nothing against or negative. But the one more study that we should do, it's actually to do a trial now using low and high acylated anthocyanin, and then see if that holds true.
Do you recall how these cultivars measured up against the sugar moieties high or in low versus high anthocyanin?
They were contrasting for not for acylated anthocyanin. They were contrasting. Yes. Yeah. Now another thing that my collaborator Colin is trying to get to, the student that was working on the project, she left, and she is trying to mine the data. So it's not easy. So when you ingest blueberry, the anthocyanin that you measure before the digestion, they are not in the circulation system in the same chemical structure as they are in the fresh blueberry. Okay, no way. So there are residual chemical structure left over in the blood system that are targeted. So then it's trying to see if we can find trace of acylated or non-acylated anthocyanin to see if those are the majority in the serum analysis. Thank you.
All right, we have a break. It's going to be a little bit truncated, so we need to be back in here.
As I was thinking with that recent graduate history in mind of what to talk about today, I wanted to be conscious of not just giving another cryptic industry talk. And so I really zeroed in on thinking about the thought process behind design breeding and sprinkling in a little bit of career path context as well. So that is what I'm going to be talking about today. Might shift to. I think I can do this. I'll just look here. Thank you so much. So just to give a little bit of background, I grew up in upstate New York in the greenhouse business, and I had my sights set on being a farm manager. So I went to a really small technical applied undergrad, and it wasn't until the very end of my final semester that I took a plant biotechnology class. in a tiny basement with 2 other students, and I discovered plant breeding. And I really fell in love with this intersection between the theory and the applied aspects of plant science. And I went on to pursue this discipline as a research technician, working in small grains at Cornell, before going on to a PhD program in the integrated program of plant breeding, genetics, and biotechnology at MSU. And I joined there at a perfect time because I was able to be part of a project focusing on converting potato from a tetraploid clonally propagated crop into a diploid self-compatible seed propagated crop. And just as I finished up that program, I joined Bayer in 2021 as a open field pepper product design breeder. And that's a little bit of a mouthful of a title that I hope to demystify today in this talk. But my primary role in this, my primary goal in this role is to bring solutions to customers. And I have a little bit of a schematic here. Is this, do you have a pointer here? Ah, I do, okay. To bring solutions to customers. And here you can see how I split my time. So I spend most of my time in cross-functional collaboration, and to a slightly lesser degree, spend my time in analytics and in field work. As you can see here, this is me in Australia talking with customers and taking data in the field, and then in strategic thinking. And there's one aspect of industry that I've really come to appreciate, which is cultivating your personal brand. And I've found while working in industry that I really love being an early adopter of novel technology to drive novel genetics faster. And that's in keeping with one of the principles that I tried to guide my life by, which is a quote from Eleanor Roosevelt, to do one scary thing every day. And that can be as simple as talking to somebody new, or it can be joining a CrossFit training camp in Austria, which I got to do last year. So as an early career plant breeder in industry, I want to tether my talk today about the journey of a product development in Bayer to the journey of a young plant breeder learning how to navigate a global organization. So we're going to start talking in broad strokes about plant breeding in Bayer, and then take a deeper dive into a case study of harvest mechanization. Often in grad school, we're so focused on that end goal, which is to land the dream job, that the elements of what a successful transition into that dream job look like become secondary or even tertiary considerations. And there's a couple key skills that are important for success in industry that are not really reinforced in grad school. In grad school, depth of technical knowledge and end-to-end process ownership are not just rewarded, but they're really essential for success in your program. You have to be a content expert, and you have to be a content expert in so many things. While in contrast in industry, you are much more successful at being a team player. And so those team-based skills are much more reinforced. So instead of having to carry the entirety of a project, you're asked, how can you influence a team or even a team of teams to implement a strategy? And it's actually the fact that your ability to optimize the opportunity in a global organization depends to a huge extent on those interpersonal skills. So there's a couple pillars that underpin our breeding impact in industry, and your interpersonal skills drive a synergistic effect between those. So for instance, if I'm at a company and I have access to amazing genetic resources, and I have phenomenal coworkers with whom to brainstorm, and I have state-of-the-art resources, but I don't have the communication skills, I will fail to leverage all of these pillars to have maximum impact for our grower. And in order to stay on top of these development skills, we have to be dedicated to a continuous improvement in personal development. And that can be kind of modeled as sequential and overlapping personal growth curves. So first we start by identifying a set of development skills. And for me, when I first started, one of the areas where I really needed to improve was in delegating tasks. So my tendency was, if I wanted to get a process done well, I thought I needed to do the whole thing from end to end. So if there was a trial that needed to go out, I was preparing the entirety of the list, all the way down to the inventory BIDs on the seed packets. And this was really inefficient, and I wasn't utilizing the team atmosphere. So I took small steps in this initiation phase to identify small projects where I can start implementing this new skill. So building a database that maps our genotypes to our trialing partners and enables our other teams to be autonomous is one example. And it's important to note that when you're on this curve going up the growth phase, not every time you try to implement a new skill, you'll be successful. But the outcome of building a new way of thinking and building a new set of relationships is what's more important. And just as you reach this apex of the growth curve where your new skills are becoming second nature, that's when it's time to initiate a new development skill. And in this way, you can stepwise improve your impact and your effectiveness. And as I go through today, I'll be showing in some little red sticky notes key developmental goals that I found were really useful in plant breeding applications in industry. So let's go ahead and look at what product design breeding looks like in Bayer. So here I'm showing 4 main functional teams that make up the breeding pipeline in Bayer. And we have product design breeding, product development breeding, market development, and then our commercial team. And the biggest take home here is that this is really a big feedback loop. And the process starts over here on the right with our customer-based market needs. So they're directly informing what our breeding targets should be. And then we have the product design breeders who sit over here, of which I am one. And our goal is to take these market needs and conduct population improvement and line development. And we develop these new elite lines and place them in genetic heterotic pools. And we hand that off, hopefully, as a tidy package to our product development breeder coworkers. And their role is to then take these elite parents and find the best combinations to create new hybrids. And then those new hybrids can be tested in a variety of different trialing locations. And that data that they generate comes back and feeds back to the product design team to inform parental performance. The product development team then hands off elite products to market development. And you can think of market development as sort of akin to a research extension position. So they sit in between breeding and commercial, and they do large-scale trials with our material with customers. And they can help advance the final products, but they can also give that really prescriptive environment and nutrient management recommendation for our products to the customer so that they have a recipe of how to grow this new product. And throughout this process, our number of pre-commercial products are decreasing, while at the same time, we're increasing the amount of tests and trials and customer feedback that we have. Our vegetable segments are super complex, and our customers can sit in 4 different global regions and all throughout the value chain. So we're thinking about the complex needs of, say, a smallholder in China, or perhaps a processor in California. And blocky pepper, which I work on, is a really good example of a hyper-fractionated market. So we're breeding for 3 different environments. So blocky pepper can be grown in the greenhouse, in the glass house, or out in the open field. We have a range of colors, only some of which I've shown here. We have what we call material class. So that would be, what does that pepper look like? Is it truly a bell pepper where it's just as long as it is wide? Or is it a little bit longer than it is wide? And that would be a half long. And then we also have crop duration. So something that's grown in the open field only has to persist for about 6 months, whereas our protected products need to be in the greenhouse for about up to 18 months. And you can think of the environmental demands on one plant that has to live for 18 months through hot and cold cycles and through different pest and pathogen pressures. So if we had a different breeder for every one of these scenarios, we would really quickly become fairly siloed. So how do we avoid that? By decoupling germplasm improvement from product evaluation. We can at the same time be driving genetic gain quickly and also characterizing our products in the target environment. So we see that product design and product development, we're both working toward the same breeding targets, but we have rather quite different mindsets. So the folks who are in product development, they're focused on what traits do we need in this product, and then how many trials can we characterize it in so that we really understand this specific combination. Whereas over in product design, we have the latitude to zoom out and think at the population level, which allows us to do things like think about diversity maintenance and rapid recombination and genomic selection. And we're divorced from the need for a specific product characterization. So in product design, we're building this genetic toolbox to anticipate emerging product needs. And one of the foundational skills of a product design breeder is this ability to come up with a new product concept, and we call this product concept ideation. There's a lot of buzzwords, but I think this is one of the most important and one of the most exciting human elements that we still have in plant breeding, because it is a very human characteristic to be able to take all of the unique market needs and synthesize a completely new plant that we've never seen before, that's just in our mind's eye, and then to decompose that into what are the different traits that we need to make that come to fruition. So I think that's a very good case for job security for plant breeders, and it's a very fun space to live in. And once we've identified what our product concept should look like, next we have the equally complicated task of looking for the genetic diversity that can fill those gaps. Once we've sourced that diversity, then we create informative populations, and ultimately we deliver those uniform lines that we hand off for routine breeding to our development partners. So I've talked at a very high level about these elements of product design, and now I want to take a case study to take a little bit of a deeper dive into what those product design concepts look like. And I'm going to specifically talk about a project that I'm managing that's transforming pepper into a machine harvested crop. And pepper has quite a few touch points. If we just look at this photo of a commercial production of some cubanelles, we can already see evidence of all of those touch points. We have plastic mulch, under which we have drip tape that needs to be laid. You can see we have stakes and trellising, which are keeping these peppers up. Not only do we put those out at the beginning of the crop, but then as the plant grows, we continue to add more strings. We have big gaps between our beds, which is areas where we need hand weeding. And then as you can see, we also have the actual act of hand harvesting. Our first step in the product concept ideation is to come up with a project justification. Why are we pursuing this idea? What value do we think this will bring? And what problem are we solving? And in this case, there's a couple key pillars and drivers for this project. So we know that the labor force is decreasing in areas of pepper production. And it's kind of a staggering number that we have a drop in US farm employment from 1948 to 2017 of 81%. And if we pair that with the fact that at the same time, our labor costs are increasing and our produce prices for the consumers are staying about the same or about flat, we can start to see that that profit margin for the grower is really getting squeezed. And then we add on the fact that pepper is a complex crop to harvest. So we already mentioned that there are quite a few touch points throughout the growing season, but also the pepper can be hand harvested in the open field environment up to 8 times in a single cycle. And we know that automation of harvest is really tricky. So a lot of the automation innovations that we see on the market right now are for these pre-harvest activities. So you see a lot of innovation these days in precision weeding and precision planting. But harvest really seems to require that intersection of improved genetics with an improved automation solution. Once we have a project justification, the next step is to scope our project. And it's tempting to set too narrow of a scope because we want to manage our expectations. But we run some risks if we set a very narrow scope. So if we say we're only going to focus on jalapenos in California, for instance, it might be the case that there's insufficient diversity in this target's scope for the traits of interest for us. It might be that we're creating possible redundant efforts in the future. Maybe there are quite a lot of parallels between jalapenos and other peppers that we're interested in, and we're missing that by being too narrow. And maybe the worst possible situation would be that we have an inability to pressure test related markets in this POC. So just to give a little bit of a concrete example to this, in California, peppers tend to be grown without stakes and ties on bare dirt. And in that scenario, we have quite a lower barrier to adoption from a cultural standpoint for machine harvest. You know, the grower doesn't have to rethink how he or she manages peppers. You could, in theory, just run a harvester over that existing setup. And the plant architecture is different too. Those plants are used to standing straight up and tall, so they have a different erect architecture. Whereas in the East Coast, we see staking and tying much more common. We see double rows of peppers, and those plants are used to being coddled, so they're ready to fall all over the place. If we take away their trellis, we'll end up with a very prostrate pepper. So if we only test in California, we miss the opportunity to pressure test those potential challenges to our innovation. But if we swing too far the other way, and we take a very broad scope, we have challenges there as well. We could lack the agility and speed to bring something to market. And also maybe we're only making incremental gains across a lot of different types of peppers with no true breakthroughs. And for me, the big learning in this process was to be very willing to seek diverse input, both in the company and out of the company, and to be really comfortable with changing my mind. I think for me, I always want to have a plan. I want to have an airtight plan that's ready to go. And in this process, this very creative process, it's important to acknowledge that your first idea might not be the best idea. And that by seeking diverse input, you can arrive at the best solution. Which sounds easy, but in practice is very hard. So next, we have the phase of a landscape analysis, which is just a term where we set the definition for our idiotype. What is this plant actually going to look like? How is it going to be harvested? And by whom? And in this process, we have to balance the value to the customer and the value to the company in our case, with some technical and logistical hurdles. So generally speaking, the more disruptive the innovation is, the more the opportunity, or the greater the opportunity to bring impact to the grower. But we also have to think about the disruptions that these cause to the daily life of the grower. So for instance, if we propose mechanical harvest, are we injecting a machinery that's too expensive or too cumbersome for some of our grower customers to acquire? Thinking about agrisystem dependencies, we'll be changing or playing around with row spacing, with in-row spacing. Will they need to have a labor crew throughout the entirety of the season? Will they rethink their entire labor strategy? And how will we partner with them to help them through that? And then technical obstacles. Will we find the traits that we're looking for in our germplasm? Will the automation companies progress at a similar rate so that we have a solution to deliver? And for me, this was an opportunity to grow my network. So I met with automation companies to understand, what does their landscape look like? Where are they, what do they have commercially available now? And where are they thinking they want to spend their R&D money in the next 10, 20 years? Most important is meeting with growers, understanding what is their appetite for change adoption. What do they see as the pain points? How badly do they need this solution now? And then meeting with trade organizations, which is a great way to magnify the voices of some growers that we might not have an existing relationship with. And then internally, we have teams that are dedicated, teams like strategic markets, consumer insights, and of course, sales, to help us once we come up with a product to think about how would we usher that through all of the phases of development to actually physically bring something to the customer. And it can be beneficial to pursue a couple of different idiotypes in tandem. And my take home here in the sticky note is to engage partners early. So by bringing in multiple different automation solutions or multiple different types of growers, we can start developing a portfolio of genetics that's robust to many different types of solutions. So here I'm showing we can think of a couple different potential solutions for pepper. So we could have a time system similar to blueberry, which is tickling the fruit off. We could have a helix system that consumes the whole plant and then strips the pepper off. Or we could have a fully robotic system governed by AI to hunt and find those individual fruits. But each of these need to be decomposed into their specific traits. And when we do that, each of these idiotypes that match these scenarios, we see that there's actually some very common traits that are a through line between all these different ideas. So all of these different types, they need some manipulation of maturity, mostly trending toward a concentrated uniform maturity. We need a manipulation of root architecture. We need to incredibly rethink branching architecture, and we need to alter fruit characteristics to make the fruit more resilient to this new mechanical stress that we're putting on them. So when we have a trait list in hand, now it's time to source genetic diversity. And we have several resources at our disposal that I've arranged here from an internal standpoint in preference. So our material that's in the breeding pipeline already, that's going to be our most elite material. If we don't have the variation we're looking for in our elite material, we can head to trait discovery. They're a team that takes wild genetics and integresses them into our elite material. If it's still too novel for that, we do have a gene bank. So we work with the USDA and have our own copy of the USDA gene bank, as well as our internal gene bank. And then if it's still too novel, even yet, we have the opportunity to pursue an external collaboration or partnership. But regardless of which genetic solution we're pursuing, we really need to think strategically about the screening environment. Ideally, we would want to screen in the target environment where we think this product will take off. But a lot of our novel genetics don't have the preferential alleles for some killer traits. So thinking of disease resistances, a lot of our novel traits won't have those disease resistances that we've come to need in our trialing markets. So we have to get a little bit more creative, sometimes trialing some things in one location, some things in another location, perhaps correlating between protected environments and open field environments. When we're making our phenotyping plan, we want to look first at the low-hanging fruit that we call pipeline traits. This is just data that we collect in yield trials, and we collect a ton of data in yield trials. And for pepper specifically, the lion's share of all of my yield trial data comes from a digital phenotyping platform. And this is an image-based platform. So all peppers get harvested and put on this conveyor belt of this adorable little machine. And they get run through here, they're imaged in this image box, and then we get a weight on the other side. And this allows us to detect individual objects and then quantify shape and color and size traits. And from those component traits, we can build models to predict fruit quality and fruit maturity, which are really important aspects of this particular product concept. If we don't have the trait that we're looking for in our current set of protocols, our phenotyping protocols in our yield trials, we can then leverage data that's collected by other internal teams. So one really great example of this in this particular project is the plant variety protection program. They sit between breeding and commercial, and they take super granular data on all of our advanced hybrids and their parents. And so some of the benefits of this data set is it's a huge historical data set. They use very large plot sizes. They have a very rigorous QA/QC protocol. And they have, as I mentioned, a very granular level of trait collection. So all the way down to things like how long is each leaf and what is the particular shape of that leaf. So in this project, we were able to leverage data, historical data that they collected for the exact novel traits we were interested in, instead of having to create brand new protocols and train folks how to take those protocols and go through the growing pains of learning how to phenotype these new characteristics. But ultimately, we want to measure machine harvestability. That's what we're trying to do here. And at some point, you need a machine to link those traits, those differentiating traits, to machine harvestability. And there's a couple different ways that we can go about mechanization phenotyping. If the automation already exists in the market, we can go out and buy that machinery and bring it in-house. And this gives us the most amount of flexibility and autonomy. And the only main downside is that then we've really hitched ourselves to a single automation solution, unless we have the capital to buy multiple machines. If we want to distribute our risk and our interest across multiple machinery, we can partner with universities or with growers that might already have this machinery. But then we have a trade-off in terms of the flexibility and the data collection aspect. If the automation doesn't exist yet, it is imperative upon us to then go out and find an automation company and perhaps form a strategic partnership with them where we develop the genetics in parallel with the automation so that we have a tailored solution at the end. But regardless of which solution you pursue, it will really be a test of your ability as a breeder and your powers of persuasion to advocate for this method, because each has a very different resourcing need and different strategic implications. So once we have our traits and our phenotyping strategy and our germplasm, we're finally ready to create some informative populations and ask some questions that will help us get to the next step. We can characterize variation for the differentiating traits and compare them to our targets. Sometimes those targets are known by some previous literature, and sometimes we're making those targets up in a sort of arbitrary space, which also feels uncomfortable. Examining relationships between component traits and understanding those implications for breeding is an important aspect of this stage. We also need to examine the relationship between machine harvestability and those component traits, examine the trait genome relationships, and then prioritize the donors that we want to use that are most aligned with our elite target markets. And the thing I want to say here is that notice how advanced we are in the process and in this presentation before I've gotten to these questions that we would associate with traditional breeding or traditional quantitative genetics. And all of the fundamental activities that took us up to this point where we get to do the breeding really rely on those interpersonal skills, the ability to involve the right stakeholders, the ability to grow your network, the ability to influence and persuade. Now we come to the point of population creation. We will be starting by creating new crosses. Then those will be phenotyped both for the differentiating traits and our novel traits. We'll work with our internal teams to develop molecular markers and molecular tools. And we'll finally have prioritized populations that can then be spun off for rapid inbreeding. And these inbreds that have the novel trait integrated into our elite germplasm are first placed in a genetic pool and then assessed for their parental performance for what we would call those pipeline traits, our yield and quality. And this is a very key decision point where we need to partner with our development colleagues, with our marketing colleagues, and with sales to understand what's the maximum penalty we're willing to accept in yield and in quality in order to deliver this disruptive innovation on the timeline that we're interested in. And even though I've been talking about a project in pepper that's in progress at this point, these are two brassica-related projects that are commercially available to our customers that are the output of a very similar process. And here we have mechanization solutions for broccoli called high-rise broccoli and for cauliflower, Curtifex, that are tackling the same kind of solutions. How can we bring a mechanized and labor-saving solution to the customer? So the broccoli solution has a taller head and fewer leaves and uniform maturity. And the Curtifex is the highlight of Curtifex, as you can see on the right-hand side, which is Curtifex compared to a traditional cauliflower, is it's that super bright white. And it stays bright white throughout the entire season. So the grower doesn't have to spend time and money and labor putting expensive netting over the cauliflower to keep it as bright white as he or she possibly can. So I hope that in this time I was able to shed a little bit of insight into the process and the experience of a product design breeder. And I would welcome any questions that you have.
Emily, excellent call. Question on the pepper that's grown for 18 months. Why is it grown for 18 months and where is it grown for 18 months?
Those are great questions. So that's mostly in semi-protected culture in Mexico. It's grown because they like to have the stability of a constant slow supply of fruit. And so that to them is better. They're not working with a huge amount of land, so they can't do staggered sequential plantings. And they appreciate being able to have continuous production throughout the year.
Hey, Natalie, thank you so much for your talk. It was excellent. I was just curious, as someone who's not in the plant world, I'm a fly geneticist. The Curtifex cauliflower, is the advantage of it being bright white just an aesthetic thing for the end buyer, or does it help people or machines see it more clearly?
It's both. So it is a consumer preference, but then also it helps if you're machine harvesting, it helps with detection. And then if you're hand harvesting, if you've ever been out in a brassica field, the leaves kind of lay all over, and it's a very mind-intensive process to be constantly on the hunt for those heads. So it helps with labor reduction as well as improvement efficiency.
Awesome, thanks so much. That makes sense.
Hi Natalie, first of all, great talk, and before I start, I just want to say that I'm all for harvesting using machines, but have you thought about what would happen if everything is harvested with machine? Because you'll be losing a lot of jobs, and will the federal government allow it? And if it's cheap, anyone can use it, all the farmers will start using it. And since you said you've talked to a lot of growers, but you haven't really talked to all the laborers. So, that would be a big problem because we'd be losing a lot of jobs. What would you do in that case?
I think that's a really great point. I think that the answer lies in diversification of crops. So I think that the growers that I've spoken to would be interested in, this would allow them to invest and pursue other crops that might have more of a manual input on a smaller scale. But I don't have a perfect solution for where those jobs will completely be lifted and shifted to, and the livelihood of those folks. I don't have a wonderful solution, but it's an important question.
Now we back here. That was a great talk. I really enjoyed that. And I wanted to start, ask the question where you started, you know, with your grad student experience, carrying the world on your back. If you had to recommend how you would structure graduate programs, you know, to be more team-based, more integrated like that, do you have any recommendations? So that's a question A. And the question B would be, do you envision the need for micro-credentialing and lifelong learning in your current position?
I love those questions. To the first question, I think that it would be important to build into our aims for graduate school some developmental goals as well. So, you know, we normally have 3 or 2 or however many specific aims in a graduate program. And I think it would be important to, with intentionality, add in developmental goals to that based on what your desired career trajectory is. I appreciated that my program was a very integrated program. So my graduate program had some field work, it had some lab work, it had genomic work. And that does force you to do a little bit more cross-functional collaboration. But you're learning on the fly. It's not built in with intention or followed up on by your committee members as a general rule. Some committee members excel in that realm, but it's not baked into the requirement. In terms of continued education in the workplace, I do think that is really important. I think that at a bare minimum, it's important to be paired with a mentor who can help keep you accountable in addition to your manager. Whether that takes the form of accreditation or not, I don't know if that's paramount, but having accountability to make progress in these developmental areas, I think is at a bare minimum, a mentor is a must. I hope that got to your question. Okay. Does the pepper germplasm have well-defined heterotic groups? That's an exciting thing I need to work on as a design breeder. That's all I can say.
Speak to like roughly how big the teams are that you're working with for like, is it you deciding to focus on California, or is it a much larger team that's kind of driving the get pregnancy?
We have an innovation division, and that's made up of breeders. So there's 6 other pepper breeders that are focusing on different market types and some different regions. They're involved. And then we also, as I mentioned, those different functional teams, we have at least 1 representative across the entire pipeline. But the team itself is on the order of 10 to 20 people. The actual group that's making final decisions is much smaller, but folks that you're interacting with on a frequent basis is about that size. Thank you so much.
All right, wonderful talk, Dr. Kaiser. So we are approaching the lunch hour, but before you get up, there's just a couple little things to take care of. So we will be doing a group photo right after we leave the room. Kristen, where are you? Kristen, right here, blue-gray shirt. I'm a little bit colorblind. I think that's blue-gray. Kristen's going to lead us out to where we're going to take the picture, and she's going to take a picture for us. So we'll do that right away. And then after the photo, lunch will be in the room to your left down this hall. You'll see it. There's box lunches in there. That's also where we're going to have our poster session. So if you have a poster and you haven't set it up, do that right after the photo. Austin, my secretary. All right, everyone, the time is 1:30. It's time to take our seats. Austin will be introducing our next speaker. Thank you, guys. And just as a reminder, the voting on posters will close after our next break. So please vote if you haven't done so already during that time.
Okay, good afternoon. Our next speaker will be Dr. Patrick Cumby. He's the Director of Product Development at ArborGen. He's been involved in forest research and development for 23 years, has largely focused on planning and implementing accelerated breeding programs in loblolly pine. Before his time at ArborGen, he was a research forester for Weyerhaeuser and also worked on staff with the Cooperative Tree Improvement Program here at NC State. In his current role, he manages product development in the United States and Brazil, including breeding, seed production, and genomics. He also has a PhD in forestry from NC State and is an adjunct assistant professor in the Department of Forestry and Environmental Resources. Let's please welcome Dr. Patrick Cumby.
Good afternoon. Thanks for the opportunity to speak today. And before I get into the slides, I'll just say thanks to my team, all the work that we do. We have a small team at ArborGen, but we get a lot done. So I just want to make sure I acknowledge my colleagues and teammates for all the work they do. I'm thinking about what to present today, particularly with trees. It just strikes me how long it takes for us to bring something to market. And so just thinking about what we do as plant breeders, that our product is always in the future. So trees really kind of magnify that when we look at the timeline to bring something, to develop something, bring it to market. So, if we have a candidate tree today that we're going to bring through the breeding program, or we have a population that we put together, ultimately it takes 25 to 27 years to get that product through the production pipeline and out to a customer. And then once the customer has it, the actual product that they get in timber is another 25 to 30 years. So many of the trees that we're selecting this year in the next month or so for our breeding program won't actually get to a sawmill to actually make a product for 50 years. So just thinking about the theme for this this symposium, a lot of the both human and plant components are they're integrated into what we do. Tree breeders tend to be very risk averse. We're conservative. We want perfect results before we move forward. We also have a lot of different components that are factored into the program along the way, looking at pest and pathogen resistance, managing diversity, looking at long-term adaptability of our tree populations, looking at financial returns for our customers, and managing risk. For Southern Forest, just as a little bit of background, there's over 200 million acres of forest land in the Southeast. Most of the time, it's one of the top 10 manufacturing industries in the southern states. Of that 200 plus million acres, there's about 37 million acres of forest plantations. And that's really what we focus on as far as our breeding program. There's a lot of money that comes from these products globally. We have more than 1,600 mills, so it's a large operation, or a large industry across the Southeast. And just thinking about what a forest property looks like that one of our customers may have, that is really kind of mixed use. And so some areas that land may have more of a conservation focus. Some may have a timber production, such as the pine plantations in this picture, which again is what our breeding program works on, although as a company, we probably offer more than 50 species each year for customers to purchase.
So just a little bit about ArborGen. We actually started in 2000, and it was a joint venture between 3 forest products companies. The focus was biotechnology. And so they wanted to build a platform of being able to clone trees and then do genetic modification. And so that was really the goal and focus for the first 10 or 12 years. But as that more or less faded out, we also acquired some seed production capabilities and nursery capabilities along the way. And so breeding became much more of a focus of what we do. So we operate in North America and Brazil now. A couple of years ago, I would have had a map of Australia and New Zealand, but that operation was sold about 2 years ago. And so we produce roughly 300 million seedlings in North America each year and about 100 million in Brazil. Our footprint covers the Southeast. So this is really our operating territory. And this is more or less what our product looks like. So just thinking about how we produce seedlings, on the left side of the slide is a bare root nursery. That's the majority of what we produce. These trees are grown in sandy soils. They're lifted, bare root, packaged up, and shipped out to customers. And on the right is a containerized nursery where there's a plug that's actually grown in popularity the last few years. But these are the 2 main vehicles to deploy our genetics. A lot of times what we work with is kind of on the opposite end, thinking about customers, is understanding that there are genetic differences in trees. And so you may have a customer who has property. They do different things. They may grow crops. They may have pine plantations on certain portions of the property. And they may pay close attention to what seed they grow for a row crop, and they may not care at all or even think about genetics in their trees. And so a lot of times we have to work hard to distinguish and educate customers on what their options are with genetics. And it really comes down to they look into the forest, they see things that all look the same. They don't have a discriminating eye. And so, you might look at a pine cone, you might look at a seedling, and you think all of these things are the same. But ultimately, it comes down to a product. And so for a long time, this is the focus of what pine plantations produce. This is pulpwood. So this is going to go to a pulp mill or paper mill. Has a value of somewhere on the order of $2 to $10 a ton. Or you can have a solid wood product that has a much higher value, $25 to $35 a ton. And this has become the emphasis of our breeding program, and is really kind of the driver for why we put so much effort into breeding and seed production that I'll share today. For forest trees, so this is a chart that has a little bit of age on it now, but is a nice graphic just looking at how we put silviculture and productivity and management together with genetics to look at potential wood production. And so this is in tons per acre. And so you can see in the last couple of bars there in 2000, 2010 and beyond, there are some growing components there with tree improvement, which would be traditional breeding, and then clonal and biotech. And so this was really the expectation that transgenics or clonal forestry was going to take over, have a significant addition in productivity. And that really hasn't happened, although we have achieved these productivity levels. It hasn't come through biotechnology. It's really come through traditional breeding and good forest management. And so some of my colleagues extended that 2020 bar there that I've got highlighted and covered up the top was a bit speculative in where we might go. And I still think that is a question mark for us as to how far we can push genetics to enhance productivity. But again, at this point, it's coming through traditional breeding methods, not through biotechnology, although I think biotechnology has a lot of opportunity that we should continue to explore. It's not made it to the market yet. So For Lottolite pine breeding, this is just a snapshot of what our breeding and testing and selection process looks like. So we work with populations in different regions and go through the same process, similar to what we do with our cooperative programs that we're a part of. We have 4 key traits that we look at. So wood production or growth. We have a native disease, natural disease, fusiform rust, fungal pathogen that attacks the trees. We're working to straighten the stem of the trees for better products and reduce forking rates. Both of those have a large impact on the solid wood products that we produce. We have more or less 5 different provenances or regions that we work with. We assign counties to those, so you see discrete lines, but really there's a gradient there. Mostly that's driven by winter temperature. So as we move north, we have to have cold hardiness. As we move to the west, we have to have drought resistance. And so most of that adaptability is managed within those populations that we work with. Our program is built off of working with the cooperative breeding programs. So there's one here at NC State, the Tree Improvement Program. There's also one at the University of Florida and one at Texas A&M. And so those are our platforms to work with across these regions. We've made a big effort on control pollinations to produce full sib seed. So this is really the driver for what we do in our breeding program. Here, I'll walk through some detail. This is what I really wanted to emphasize today, was just the effort that it takes to get advanced genetics out in a forest tree system is not anything that's fancy. It doesn't come through a lab. It's not complicated. It's really brute force at this point of combining 2 great trees together and control pollinations. And so this is just a snapshot of what that process looks like. But this is when I talk about MCP or mass control pollination, this is a commercial scale process to make full seedlings. And really the reason we go to that effort, try to highlight in a couple of pictures here, but this is a picture where there was a traditional open pollinated family. So we know the female, the mother, but we don't know the pollen parents of this family. But if you don't select for appropriate trait performance, you end up with something that's really not ideal. So this picture just kind of highlights some of those problems where you've got trees that are not straight. You got a lot of gaps in the stand, so you're missing the opportunity for biomass production. And so it's really just not ideal and not efficient. And this is compared with, one more slide there of severe rust infection. And so you just lose the opportunity to produce a tree that has the characteristics that you want. That's contrasted with a superior cross. This is a full sib cross, but you've got a lot better survival in the stand. You've got good stem form and good growth rate. And so we've combined those traits. And this is what that control pollination process offers our customers. So this stand has been thinned, so there's a partial harvest at mid-rotation to space the trees out and allow biomass allocation on a reduced number of stems. So this what ends up being the final product, the trees that are harvested. And so when you have good quality and good growth, you can space that out and grow more efficiently and produce a higher value product. So again, just highlighting what does it take to make that, to make a cross. So a lot of times we focus on that first part of breeding, testing, and selection. And we've gotten that down. That itself used to be probably on the order of 20 to 25 years. So that's now down to somewhere in the order of 12 years. It could even be sooner if we're successful at every step. But and a lot of times as breeders, that's where that's our world. It's that breeding, testing, selection. And so my colleagues that are here with the tree improvement program, as soon as we're done with that 12 years or even before, they're thinking about the next round. What's the breeding strategy for the next cycle? What's that population going to look like? On the commercial side, from my perspective as a member of the co-op and as a seed producer, those next steps really move into product development and commercialization. And so we then have to establish a seed orchard and seed production, and that takes quite a bit of time and effort. So we want to be right in the trees that we select. We are looking at tools. I'll highlight some of these later on in the talk, but looking at genomic prediction methods to shorten that development cycle. We're looking at fingerprinting and purity testing in our crosses to ensure our product is what we say it is. There are places to use technology, but we're really relying on traditional breeding. Just a few pictures on what our breeding efforts look like. So usually it takes 7 to 10 years for a tree to reach maturity and start flowering. And so traditionally we would have grafted a tree at ground level, let it grow, produce flowers in the time, whatever time it takes. Maybe use some hormones or auxins and speed that up, but it would have taken several years. And now this has become a standard of the last 25 years of top grafting, where we take a selection and we top graft it onto an existing orchard tree that may be 30, 40, 50 feet tall. And in 1 year, we can get flowers, it'll stimulate flower production. 1 or 2 years, we get flowers produced so that we can do that breeding in 1 or 2 years instead of 7 or 8. So that's allowed us to shorten that cycle, and that's pretty much a standard practice for us. Let's go into field trials. These are typically 1 to 2 acres in size. We spend a lot of time thinking about our methods, and so we look to our university colleagues on experimental design recommendations, and we try to put things, best methods into practice. So this is just a quick description of what that looks like. Incomplete block design. We typically replicate over 3 to 5 sites. Each site has 10 to 15 replications per family. And so we're really looking to test about 100 families per site. And then we would measure this probably at age 3 or 4 years, and then again at age 6, and do our breeding value calculations to make selections. So we spend a lot of time managing pedigrees, looking at our field trial designs, and trying to improve this process. So we have to set some guidelines on what goes into our full sib production. So if we're going to make those control pollinated families, we're looking closely at the data to make sure the value proposition is there. So we don't spend a lot of time making a control cross that's equal to something we can get as an open pollinated family. And so we look at those 4 traits: our growth rate, rust resistance, the stem straightness, and forking. We put that into a more or less a basic index, and we try to look at the best combinations that our families, our crosses, can offer. And so this just represents looking at those commercial crosses. The value proposition and trait performance is better than what traditional families offer. And so this is kind of our threshold. Our minimum performance needs to be there so we can justify the product. And if we're doing that correctly, then as we move up, this is just an example of different product classes that have growth rate differences. And you can see that range from about 50% up to just over 90%. So we have a pretty wide range in volume gains depending on the product. And so as we continue to work through our program, we are now able to combine, make unique combinations that really match the traits together. And so we've been able to make some superior crosses with superior form and growth and bring those to market. So those are probably the product of a 10 to 12 year effort internally. But those are now on the landscape, and customers can acquire that material. So that went through this process. So I'll highlight what goes into the seed orchards. And so, seed orchards are typically relevant for 25 to 30 years. At 30 years, they're about 100 feet tall. It's hard to get a person up there to pick cones. So when you collect pine cones, that is a manual process. Because that lasts so long, again, we want to be sure we have the right tree going in, because we are carrying that production for a couple of decades. Here's an orchard that's being established. So these positions are laid out on a grid, and each position will be grafted. They might be grafted in rows to make full sib crosses. They might be randomized to allow for a better mixture of pollen for open pollinated material. But we spend a lot of time managing these sites. They would get drip irrigation. We'd manage the fertilization and nutrition closely, and try to get these up and running so they can produce seed as soon as possible. Just a couple of pictures. This time of year, we're preparing to focus in on what we see in these two pictures. So the male and female strobili or flowers, but we will start to bag the flowers, the females, in the next couple of weeks in most places here, and we'll prepare to monitor and collect pollen. So this is a large effort for us. I think the last 2 years we put up between 1.2 and 1.5 million pollination bags. I got a few pictures of that just to look at the effort. I showed this picture earlier, but you can see in the top left picture, we're applying the pollen with compressed air. And so each bag will get 1 or 2 shots of pollen. The target is, getting 1 to 2 cc's of pollen in that bag. And the trees, depending on size and flowering, there might be 50 bags per tree. There might be 200 bags per tree. But this is all manual process. They work with aerial lifts. I think our effort this past year was something like 300 lifts to get our bags up. So there's a lot of efficiency to work on in this process. It's all brute force. So these are craft paper bags, the same manufacturer for hybrid corn. They modify the bag size for us to fit trees, but it's a pretty simple method. We have to manage a lot of pollen, so we're managing thousands of liters of pollen to get processed, to get it dried down to a target moisture content, to get that so we can both store it long-term in the freezer as well as use it in the compressed air systems. And we will also germination test that to make sure it's viable. So all the basic things that we do in our research breeding, we're doing at a large scale to make this work. So just another picture here. This was a picture of one of our Texas seed orchards. That's 270,000 pollination bags put up in the trees to produce MCP. So that's been a growing product across the Southeast. So this is information that the tree improvement program here at NC State tracks, but we're approaching the production and planting of 200 million full sib seedlings each year. So that's probably 20% of the market or close to it. ArborGen accounts for about half of the production in here. So we've invested heavily in the deployment of full sibs. And I talked about the process is brute force. So it's not exciting. It's craft paper, drying pollen on a large scale, but it works to deploy advanced genetics. And so, if there was a nice biotech solution where we didn't have to do these things, we would love to do that, but it has not happened yet. Part of the success has been able to demonstrate and communicate to customers and land users the value of putting superior genetics on the landscape. And so we spend a lot of time talking to customers about the value proposition. Some, the customers range from wanting to go see the trees. So numbers are great, but we really want to see what the product looks like. Others want to see a discounted cash flow analysis, and so we work with a lot of different customer types to provide the information that they need. So that was a kind of overview of the effort we put into full SIB seed production. I got a few slides just to highlight things that we're working on now to try to enhance that process and incorporate new technologies. So one thing that we continue to look at is pests and pathogens. So some we're familiar with, like fusiform rust. Others, we have some forest health cooperatives across the South that are looking at exotic pests that may be coming or may be here that could be problems. And so that is something we pay attention to and participate in genetic screening trials where there are opportunities. We continue to look at genomic resources, enhancing our development timeline and breeding cycle, and also starting to look at carbon. So that's something that's been relatively new for us. So thinking about rust resistance, so this map is a adaptation to an older map, but generally highest rust hazard disease hazard for us is in South Carolina, Georgia, Alabama, and North Florida. And so as you move north out of the deep South, it becomes less of a problem. As you move west, it becomes less of a problem. But we're seeing some changes to that. We looked at the value of rust resistance a couple of years ago and what that might look like. And so it could be a significant impact if you have higher incidence on a site that you're not mitigating with genetic stock that's resistant. You could give up a lot on your potential investment. So that's something that's most people in the coastal region are aware of, but as you move to northern regions, that's becoming more of a problem. A couple of more recent publications have, with different analyses, identified, up in North Carolina, northern North Carolina, southeastern Virginia, there's an increase in rust incidence with some warmer springs, milder winters. And so that's something that we have to pay attention to. So we've been able to put in some additional trials to look at rust resistance in these populations where it typically hasn't been as big of a problem. So this was just a example here where we have some check lots that range from 40 to over 70% infection. We've got some full SIB crosses, and we've got some OP families. And so we've got material from testing where we can capture and deploy resistant material in those areas if that's a concern for customers. We've also, we continue to do work with rust resistance. So this is a place where we've known for a long time there's some major genes in QTL that affect rust resistance. We've been able to identify one in our populations and confirm through multiple experiments that that's there. We put that out in pilot trials across the landscape. We probably put 200,000 plants out in 2019 and 20. And so here's an example of where we potentially can deploy a marker selected product if these results hold up. I really just have one slide on carbon, but this is it could be something that's a bit of a game changer for forest landowners, and it could be nothing as far as a financial return or impact. But there's a lot of interest and energy now on carbon credits with forest land. And so we've been looking at how genetics can impact what happens there. And so basically the baseline in these charts is kind of run-of-the-mill standard market productivity levels compared to advanced genetics. And so we can certainly capture a lot more tons of carbon through advanced genetics. This is this is a level of detail that's probably not in the carbon discussion right now. But as breeders, we know that there's an opportunity here. So this is potentially a big opportunity for landowners to capitalize on planting advanced genetics. Genomic resources, genomic selection. We looked at different approaches on genomic prediction. So these are things where we have some ongoing experiments, but it's not integrated into our breeding program yet. We did some work the last few years with a population of about 1,500, able to find some good correlations through different modeling approaches. Attempted some cross-validation, see some pretty interesting results that we're starting to work with, making selections to breed for a next generation. See if these associations and prediction models hold up. Also looking at the value of clonal progeny testing. So this is also something that really NC State Tree Improvement did very well a few years ago. We've got the capability to do this, and so we're implementing. Again, this is, I think, a great for us, a great example of tech transfer from the co-ops, in that we have the ability to do the clonal propagation and able to put some trials out and see a value. So hoping and working towards making forward selections. So typically our selections that go into seed orchards are parental backward selections or parental selections from progeny tests. And forward selections have a lot more error associated with them. They're not guarantees for genetic performance. And so we think they're scary. We stay away from them. But having the clonal data gives us a lot more confidence in making those selections. And so these are the parental selections where we have accuracies 90%, 90 to 95%. We've got seedlings in this trial that are where what we typically see making forward selections, where accuracies range from 0.5 to 0.7, and the clonal selections, forward selections, were somewhere in the middle. So we see this as an opportunity to incorporate this into our program to move faster. All right, finally, I'll just wrap up, thinking about how we move forward. So this is kind of a working model of what we think our next round of breeding strategy will be, and that we'll do a pollen mix breeding rather than making individual full sib crosses for our research breeding. We'll do it with a blend of pollen. We'll do clonal progeny testing for those increased gains, and then implement some type of genomic tool. So it could be, at a minimum, the trees that we select out of those polymix families we can use to identify and reconstruct the pedigree. It could go all the way to genomic selection if those models hold up and we see the value in it, but we'll definitely incorporate some form of genomic resources there. And then we want to move faster with our orchards. So again, that whole timeline of 25 years just to get it to the market, we'd love to cut 5 years off of that somewhere. And so there will be a strategy of moving faster with orchards so that we can not only move faster in the breeding, testing, selection cycle, but also in the product scale up, we can move faster there. So again, for us, time is money. Most of the time it's where people lose interest in tree breeding because they look at the time scale. But for us, that's the challenge to work with. So we're always looking for ways to make that move faster. So I'll end there. Thank you.
Thank you for the talk. I just wanted to ask, like you said, time is the biggest challenge. Like for us plant breeders, what are the other challenges for tree breeding?
Well, I think the changes in there's a few I can think of. Changes in the industry and the way the industry functions has been a challenge. So 20 years ago, most of these programs would have been in an integrated company. So it would be an internal breeding program that goes to their own land, it goes to their own mills. And so they can move as fast as they want. Having people understand the value of genetics was all part of the company culture, if it was there or not. So since then, we have, we're much more fragmented. So we're now a seed and seedling company that's separate from the land, that's separate from the investors that own the land. And so I think communication and education is one. Biologically, with trees, I think we're starting to see we're seeing the need to perhaps add some additional traits. I don't know if they would actually be traits we would breed for, but we certainly select propagation efficiency. So we're seeing some of the advanced selections that don't graft and propagate well for our seed orchards. We always have trees that are great in their breeding values and they don't produce seed very well. So I think seems like that is getting to be a bigger problem.
So when you're first talking about, only being able to harvest up to 100 feet, that was great use case for drones, and then you showed those pictures and like never mind. Do you foresee a future in which they start integrating mechanized harvest or mechanized fertilization, or is that just not a possibility?
I think as long as we're using the pollination bags, that's probably a challenge. I don't think, it would be a stretch for us to get there. There are some efforts to use drones to blow pollen over some crops. And so some people are attempting that. There were some attempts, I think it was called supplemental mass pollination maybe 30 years ago. But it has to be in a place where you can isolate the outside pollen. But that could be if you can put the trees in the orchard in the right location, that could be an option.
So I saw that you were storing pollen. How long does that pollen store for?
I think the data would point to you can store it for about 3 years before germination really starts to go down. In vitro germination test results will start to go down after 3 years. For research pollen, we might keep it longer if it's the only pollen collection we have for certain selection. But for the commercial production, we're trying to use pollen in 1 to 2 years. We actually have an emphasis to use fresh pollen. So our pollen processing is set up to collect pollen this season, to dry it in 2 to 3 days and turn around and use it this season without having to store it. So this kind of anecdotally, we see better results. But at least we can store it for 1 to 2 years without too much concern.
Perhaps I missed this, but that graph you were showing with positive offset, or
oh yeah, so that's reflecting mid-rotation harvest. We would call it thinning, but you go in and remove a certain portion of stems, and so you're seeing a volume production go down, and then it recovers.
Hey, Patrick, there's a couple of questions from the chat here, Carlos. One from Jen Bonner in Florida. How genetically diverse are your breeding parents in the process field? And the other one is some information about how many teams are involved in the system.
Okay, so the first question in our crossing, so within a given region, we have those 5 regions. We might use 20 or more parents in a given region to make crosses. There certainly would be half-sib relationships from one cross to the next, but the parents used in any specific cross are unrelated. For rust resistance, I think the literature would point to at least 8 to 10 QTL. So I don't know if those are all individual or separate major genes, but something like that. I'd say 8 to 10 major genes for resistance, plus some level of quantitative resistance with smaller effects in genes.
Nice talk, Peter. You mentioned challenges of, being a tree breeder. They have to, work with pine and trees. There are some challenges, but why is it cool to be a tree breeder, like advantages compared to other species?
Why is it cool to be a tree breeder? I don't know. Is it cool to be a tree breeder? I can say why I love tree breeding. So, I mean, for me, working in a smaller company, I get to do the whole process, or at least be a part of it. So we have a small team for product development and breeding, and we have a team of 8 plus myself. And so we may have specific responsibilities, but during the year, we're all pitching in to get everything done. So for me, I really enjoy the kind of seasonal changes with the work, ongoing projects. So it's not the same thing every day. I think for me at ArborGen, it's been a lot of fun because rather than being part of a larger integrated company where maybe seed production is just viewed as a cost for the wood, genetic improvement and producing new products is central to what we do. So I get a lot of questions from our CEO about what's coming next and how things work. And so we get to interact a lot. It's a lot of fun.
So maybe I can add something to that, why it's cool to be tree breeder or geneticist. And in tree breeding, we work on the populations like cattle or pig breeding. We don't necessarily, work on varieties. So we use a lot of methods from animal breeding, quantitative genetics methods. And this is because trees are different. And they are outpost species like cattle and pig. And that's why we just follow their literature a lot, a lot. But also we use experimental design. So plant breeding, animal breeders cannot do that. And that's another advantage we take from, combining the best of the 2 worlds. And we can produce big families like, and the first experience, we can get thousands of progeny. Animal breeders cannot do that. So there are a lot of nice advantages of being a tree breeder. And there are some other advantages. People also outdoors, for example. We probably have one more question. Carlos, anything in the Zoom chat there? Thank you.
Just want to say real quick before our next talk, we are vacating the lunchroom. I got all the posters out. I pulled out 3 poster tubes, but if you may have left something in there, now would be a good time to go take a look. They're going to lock us out of that room here in a minute. You have to hide it each time you share the screen. Wait a second.
Hello, everybody. My name is Grant Billings. I'm a PhD student here at NC State, and it is my great pleasure to introduce our next speaker, Dr. Jenny Clement Kobernick. Dr. Kobernick grew up in Jackson, Tennessee. She completed a bachelor's degree at the University of Tennessee at Chattanooga, where she studied biology and chemistry. After that, she moved on to her master's at the University of Tennessee at Knoxville, where she worked on cotton physiology research. After that, she went to Texas A&M for her PhD, where she worked with Dr. Steve Hay. After that, she did a postdoc with CSIRO, working on cotton breeding in Australia and Narrabri, New South Wales, Australia, where she did lots of research on fiber quality and creative ways to increase yield in cotton. After that, she moved to Auburn University, where she is now an associate professor of cotton and soy breeding, a rare instance of a dual crop breeder outside of the grasses. She has an extensive record of service to the cotton, soy, and general plant breeding communities. She has supervised numerous graduate students, and she is the current vice president of the Plant Breeding Coordinating Committee. And that's the group that came up with the idea of creating the National Association of Plant Breeders. So it's pretty great that she's serving that organization. In 2023, Dr. Koverdick was appointed to the Plant Variety Protection Advisory Board. Today, Dr. Koverdick's talk is going to be, current research. Dr. Koverdick is researching a variety of diseases that affect cotton and soybean. She's also looking at seed quality in both of those crops. The title of Dr. Koverdick's talk is Recognizing Opportunity and Navigating Challenges in Breeding for Two Major Row Crops. So, Dr. Koverdick, the floor is yours. Working on it.
Were you taking notes last night at dinner?
I see you talking.
It does count after a while.
You good? Is that working?
He's good.
We're good? Okay.
You can see you.
Can I see, is there a way for me to see it? Just because. There we go. That should work. I can work with this.
Okay. Use the arrow keys and that's what
This, okay. All right, so thank you all very much for inviting me to be here today. I'm quite humbled and honored to be asked to come talk about breeding two crops. When asked about specifically what you wanted to hear from me, it sounded like you really wanted to hear how I deal with it, more so than necessarily the science behind it. So it'll be the talk may be a little different than some of the other ones today. As Grant mentioned, I know cotton. Soybean, I've learned along the way, but I would say that I know cotton breeding probably backwards to front. So another thing that I'd mention is I characterize myself as a phenotypic field breeder. I spend a lot of time figuring out which are the best parents to use. I make crosses, I walk fields, I take data and make selections. I'm not a gene jockey. I don't do a lot of genomic work, but I do surround myself with people that can do that for me and help out. So just wanted to start with that. Agenda today, I kind of gonna give you a little bit of backstory of how I ended up with two crops, some of the resources that I was giving and then what I've acquired, challenges and opportunities, and then a couple projects where I leveraged those opportunities. And then some tips moving forward for those of you that may be assistant professors or grad students considering taking an academic job. So a little backstory, Dr. David Weaver was hired on at Auburn in '81 to be a soybean breeder. And around the early 2000s, reniform nematode was a big problem in the US in cotton. And Cotton Inc. approached him about possibly breeding for this trait, working with the nematologist. And at that time, checkoff dollars were really low. And he said, okay. So he took on the challenge. The objectives that he worked on was reniform nematode resistance, a little bit of cultivar development for yield. And in soybeans, he was working on frog eye leaf spot yield and high oleic. So starting out any program, a breeder has to determine their objectives. One of the first things that I was tasked with from the cotton community was to write the history of the cotton breeding program at Auburn. And at that time, when I started, I knew of Dr. Weaver's limited work, but digging through the literature, the USDA used to have a facility there in Auburn. It moved to Mississippi in the 1980s. But a lot of groundbreaking work with fusarium wilt and root knot resistance and the complex between those and the multiple sources of resistance that came out of Dr. Raymond Shepard's work started and originated in Auburn. And this was really due to the Tallahassee location. Our experiment station about 30 minutes away had a hotbed, a really hotbed of fusarium wilt and root knot nematode. In fact, that's the reason the experiment station was purchased. So in addition to root knot nematode and fusarium wilt and runiform nematode, Alabama has been is a hotbed of diseases. In fact, Carlos last night said, Alabama's got diseases, doesn't it? I was like, absolutely. And then coming from CSIRO and working with, it's essentially their USDA, but it's partnered with a commercial partner. And so everything that we did had to have some kind of end commercial purpose. And so I couldn't help but also focus on yield and fiber quality as well. So those have been my primary objectives in cotton. And then moving into soybeans, Weaver traditionally grew 7s and 8s. He bred 7 and 8 maturity groups. If you look at the map of Alabama, majority of soybean acreage is in the north, and now they're doing maturity group 4s followed behind wheat in a double cropping type scheme. And the only county that grows groups 8s now is Baldwin County, the far south Mobile area. And dynamics and things have changed through time to where now cotton production is predominantly in the south of Alabama and soybean production is predominantly in the north. So it made sense to kind of shift the program to maturity group 5 or 4 to 5s. I inherited a couple of high oleic crosses. So in soybeans, soybean oil, the hydrogenation of soybean oil was banned, and there's been a big push from the industry to switch over to high oleic, low linolenic soybean oil quality because it's supposed to be as good as extra virgin olive oil. So that was on the list of soybean breeding objectives. We had foliar pathogens as well. It's very similar to what's in cotton, and of course yield. So I just want to kind of give you a little picture that I didn't step into a fancy smancy program. I was given $200,000 to start up an office lab and a truck. I'm on a 9-month appointment, meaning you got 3 months that you have to secure research dollars for. And that's kind of hard for an assistant professor starting out when you don't know how much things cost and you're trying to juggle whether or not you should pay yourself or pay a technician. I had 1 year of a technician on soft money. My soybean threshers were from the early 90s, and you stuck a plant in to thresh and the beans would just kind of pop everywhere. And so trying to hold on to those seeds was a challenge. This was my cotton gin. If you notice, the back of the cage is actually a rabbit cage. And the little pan that's duct taped on is a loaf pan that's covering the gears to make sure nobody hurt themselves. Along with a little tray, an oven tray underneath to catch the seeds. So in cotton, we have to delint seeds. So if you keep the fuzz on cotton, it can't go through the planter. It'll plump up. Acts kind of like Velcro. And so you have to delint. Normally you would have several large sinks in a large ventilated area. That's the sink I was working with. I actually had a good bit of cotton germplasm start, but I did not have very much soybean when I took on the program. So I am probably, most people would say I'm a very positive person. I'm very optimistic. I'm kind of a glass half full type of person. And so I said, okay, well, what do I need? I had immediate and long-term needs. I needed some equipment. I didn't have a seed counter to start with. And that was a challenge. I recognized I needed a seed counter. I needed better cotton gin. I needed some field equipment, and the lab that I was working in was doing, kind of 2 crops, but 2 crops on a very, very small scale. Dr. Weaver was winding down his program, and I needed more space. So within the first year, I tried very hard not to use my startup, and I lobbied with other PIs that worked in cotton in the college and was able to get a large equipment grant. So I was able to put in a very tiny bit of money and get a cotton gin, a seed counter, and a printer. I took on my first student that was funded by the Turkish government. I was successful at increasing funds at both the Alabama Cotton and Soybean Commissions. And I took on my first project with the cotton, with a cotton ink core project, which I'll talk about in a minute. And I took over the FOV nursery. So traditionally, our variety testing had run the FOV nursery to screen lines for commercial companies. I took that as an opportunity to potentially generate some income for the program, and took that on. By 2020, I identified a space that Dr. Weaver had used a long time ago and was able to convince the college to give me some new, to insulate it and to put a new floor in. And I set up a new lab. I used some of my startup for cold storage at this location, and now I have a space dedicated for soybeans. In addition, we needed new equipment. We had one SPC 40, which is very slow. And as we took on new faculty members, there was more plots at the station. And if you're not in line and there's a weather event, I mean, you're at the mercy of what the station can do. So strategically, I figured out who I needed to talk to, and I convinced the college to purchase the combine and the commodity groups to rent the combine from the college. And this pretty much helped with sustainability of the program. So now I feel like I'm in a place where the cotton and the soybean programs have enough resources to maintain growth and stability through time. So a little bit about the process. Now more on challenges and opportunities. So it is a challenge to have two crops. They actually compete with each other for acreage in the state, meaning one year there may be more soybean money, the next year there may be more cotton money. There's limited resources that are available. And when you have two crops, they kind of, if you want to grow, you have to, I had to find more space. Plant diseases are everywhere in Alabama. Dr. Bill Rooney, my plant breeding 2 professor at A&M, said, you know, to be a good plant breeder, you got to know the crop. So I had a lot of learning to do with soybean breeding. I had to learn soybean production, the methodology, and even how funding worked for soybean. So I was very familiar with Cotton Incorporated, but I didn't have a clue about United Soybean Board and how any of that worked. Another challenge is if you look at the pictures, the cotton flower is on the bottom, you can see it, it's easy to emasculate, it's easy to make a cross. Soybeans are not. Soybeans, I'm going cross-eyed trying to see if I can emasculate properly. And after I do, and after I make the cross, I come back 3 weeks later and there's 3 pods that look just like the one that the cross that I made. So still, there's been a big learning curve with the soybean crossing. In addition, a pod gives you 2 or 3 seeds, whereas a bulb will give you 25 seeds, roughly. So I can make fewer crosses in cotton and have more seed. So in terms of research versus breeding programs, so I run probably 24 field trials strictly for breeding program. And that is separate than the research program. The research program looks at specific problems that we try to solve. And so I'm 75% research, 25% teaching, and running a breeding program, 2 breeding programs on top of that. There are 2 times the amount of professional meetings to attend. And at one point, I actually had 2 different diversity panels, 1 for each crop. I work with conventional soybeans and conventional cotton. I don't work with traded material. And I think I'm probably not the only one in the room that has struggled with experiment stations and management. In 2020, soybean program was pretty much wiped out by Roundup. They sprayed something they thought was Roundup, but there was Roundup in the jar. I also struggle with weeds. At the plant breeding unit, however, if I ever release a variety that's bred at Tallahassee, it will grow anywhere. It has every known disease, and it has poor management thrown at it left and right. So I'm optimistic that that will be where my lines come from. So on the flip side, I get to go after funding from two different structures, two different organizations, two different sources. I divide my labor. My students touch both crops. They get exposure to both soybean and cotton. I feel that makes them a more well-rounded student. They learn a little bit more, and I spend less money on labor. I have twice the amount of collaborations. And what I learned is that some of my objectives could be similar across the crops. So my first one that I'm going to talk about is Target Spot. Target Spot was brought to me from the cotton side. It is a foliar fungus, Cassicola. Chronospora cassicola is the fungus. It produces these lesions that look like a target, and if it hits it too fast, it can defoliate prematurely. And I started out starting to look into the research, to the literature for cotton, and quickly realized that it also happens in soybeans. I was at a soybean breeders workshop the following spring after getting this project with cotton, and this is the pathologist at Iowa State saying Target Spot had been found and is a problem in Iowa now. So looking at Target Spot in general, the yield losses were reported at 18 to 32% back in 2016. And in last year, 2022, 425,000 bushels were lost to Target Spot. In cotton, right at around 3,100 bales were lost. It is prone to fungicide resistance. So basically what happens is you drive in, a farmer goes out and looks at his field and it looks great. If he walks out into his field, he might say, oh no, I got a problem. Because as the canopy closes, it creates this warm, lush, wet environment that this disease thrives in. And by the time it's spotted, it's really too late to put a fungicide on it. And the other challenge is knowing some of the extension specialists have put fungicides on it and then they don't see Target Spot. So it's a waste of money. So there's been a fine balancing act on how to manage Target Spot. So for me, I want to know, okay, are there any lines that already have resistance? In cotton, it's documented in the literature in the 50s and the 60s, and then it just goes away. You don't see it anywhere. And around 2007 or so, it popped back up. And that's when it became a problem. On the flip side, there are resistant lines in soybeans. And here was this is publication saying that the phenylpropanoid production system is somewhat associated with it. And this was a lot of work that's been done in Brazil. Now, the Target Spot hits a lot of crops. This doesn't hit just cotton and soybean, but there has been a lot of work in tomatoes and rubber tree. And so there's some literature as to what to focus on. So the strategy I took was, okay, so Cotton Inc. has funded me a project in cotton. I went after a production grant, an internal production grant with our College of Agriculture to get a Target Spot study in soybean. So I put 2 grad students on, one in soybean and one in cotton. I collaborated with the pathologist at Auburn, Dr. Kira Bowen, Kathy Lawrence, and Ed Sikora, and also Tom Allen and Tessie Wilkerson over at Mississippi State. I attended regional soybean disease workshops and participated in Cotton Inc. Target Spot working group meetings. Now then, the people in these meetings are pathologists. So keep this in mind as we advance through these stories, that I am becoming friends with a wider network than just breeders. So for cotton, we developed a protocol to be able to inoculate it in the greenhouse. We determined that we needed 50,000 spores per milliliter to be able to detect genotypic differences in lines in the greenhouse. And we also needed a mist chamber. That's really important. And when taking it to the field and doing field ratings, we found that results from the field in the greenhouse, we couldn't really tease out fine differences, but we definitely had the tops and the bottoms kind of separate. So in soybeans, there are 13 varieties that are resistant to target spot in the Grand database. However, when you go to look how they measure target spot resistance, there's nothing there. So when we tested it, not all 13 are resistant at all. But it was a good exercise. And we did identify a handful of lines that do indeed are resistant. And she's my postdoc now. She did a protocol kind of based on the cotton. The cotton gave her the sort of the groundwork to do it, to determine how to do it in soybeans as well. So now we've got 2 separate protocols published on how to do this. But I was really curious as what's the mechanism of resistance? Because my thought is if we can figure out what's going on in soybean, could that lead us or point us in the right direction to what to look at in cotton? And so we did a genome-wide association study with 246 lines. She put them in the greenhouse. She put 2 different isolates on them at the V3, V4 stage, waited 24 hours, extracted the DNA, and then 24 hours later, extracted the DNA as well. It also did a RNA-seq, and that sort of those 2 things kind of narrowed down the amount of genes to look at. So we found 6 SNPs across both isolates. And we went from 993 candidate genes to 238 and determined 26 that are involved with the resistance specifically. In this picture on the left, A is Bedford. It is in the collection as being resistant. And then on the right is one of the lines we released long time ago, and it's highly susceptible. So we plan to take this a step further and do it in cotton. And we're really hoping to see are we going to see any similar results? And if so, where would that lead us? So that's the first project, and it kind of wrapped up. We published the GWAS and the RNA-seq this past year, or 2023. And then while that was all going on, something else was happening in cotton. And I got a call from Cotton Incorporated that said, hey, cotton blue disease has been spotted in your state. And cotton blue disease is a disease that happened in Brazil back in the early 2000s, and it wiped out production by like 80%. So we've all heard in the cotton community, we've heard of blue disease, but we're really surprised that it could possibly be in the States. So I actually have a little bit of a video, if it'll show. I don't think the audio is playing, but that's okay. So this is a field down in Baldwin County. I mentioned Baldwin County is down south in the Mobile, by the Mobile area. This is in November. So if anybody has ever seen cotton and knows about when to harvest cotton, cotton's not supposed to look like this. It's highly vegetative, and there were no bolls. And there was even some stacking of the internodes. So basically what's happening and what we've learned since then is that cotton is a perennial by nature, but when attacked by the virus, it goes back to a vegetative state. And so I was kind of like, oh no. And so well, that's coming. But a little bit of background, it's a polarivirus. I mentioned 80% yield loss. Here's a little picture of some non-resistant varieties. And it is vectored by the aphid, the cotton aphid. So what we identified in the US was a third strain of the virus. In walking one of my field plots, I noticed, so this is This is one of this is the RBTN, which is our regional breeders testing network. That is simply our public breeding lines that get entered in. I grow this trial, and I stack my reps. And so on the end of the third and the fourth rep, you notice that the second number kind of turned to red, and those are yield values. I was walking through this field taking notes, and I just noticed that everything started looking like sticks, like just sticks. There were no bolts, no nothing. And so this was the same year all of this is going on. And we calculated the average yield difference across the varieties to be a loss of around 678 pounds. We did confirm that CLRDB was there. So my role in all this, when our pathologists recognized that something was wrong with this field in Baldwin County, I called a field day. And I recognized that that is not my job, but I felt compelled because if people didn't see this, they wouldn't take it seriously. So I called a field day, and I called Cottney, I called private industry, I called the other plant breeders, the pathologist. And if you remember, I know all the pathologists. All the pathologists work on more than one crop. So they work on major row crops. They're working on cotton and they're working on soybean. And so they showed up. And then we took it a step further and had a follow-up meeting. I actually invited a grower to come and sit in on the meeting. And we just had discussions on what we knew, what we didn't know, and what we needed to find out. I sat down with the research entomologist and I said, hey, this is an opportunity that we have. Where can we take it? Where can we get funding? So to test to see if you had the virus required a PCR test that was $30 a sample. We were getting samples left and right from growers. And if we were going to do any field work, we would have to sample. So $30 adds up really quick. I pursued an FFAR ROAR grant and was successful. Followed up by the CPPM crop protection and pest management grant, where we were designated Center of Excellence status. I went to the Alabama Cotton Commission. I went to Cotton Inc. I had a meeting with the university president at that point, and I'm going to talk about this later. It's like leveraging contacts. Dr. Kara Bowen, the pathologist at the time, our president was Dr. Stephen Leith, who was a pathologist by training. So Kara had done postdoc under Dr. Leith, and so that was sort of a gateway to try to get a meeting with him. And basically, the meeting was, hey, if this field is like this widespread, it is going to be extremely harmful to the cotton industry. And we asked him, what are other funding agencies? What's other pots of monies that, as an assistant professor, I don't know about? Then went to a regional cotton commissions. I drove over and met with the head of the Georgia Cotton Commission and the head of the Mississippi, well, I guess the Delta Council, which is head of the Mississippi Cotton Commission. And it was more or less like, hey, we have this issue. Please support the people in your state. And by 2020, we were able to get federal funds to research this problem. And one of the reasons why I think we were successful in achieving that is because in 2019, when we realized there was a problem, I called up the people I knew and I said, hey, this is a virus. Viruses have a genotype effect. They have an environment effect. Can we please plan a trial of like 3 or 4 varieties in all these different locations and see what we're going to see as symptoms? Are they going to be the same thing? Are they going to be different? Are you even going to have CLRDB in your area? So I was able to get this put in 15 locations, 10 other states, and I asked them to do it for free, and they obliged. And I will say that the reason I think that they obliged is because they were at that field day and they saw how bad it was. My role with this was screening germplasm. So I took I have screened greater than 1,500 lines over the past 3 years. I have pulled things from the cotton collection. I was given the diversity panel, to the cotton diversity panel to stick in this. We found 40 lines that that were not infected in 2 years in both reps. We ran a leaf disc assay on there to see if it could be infected. It can be infected with the virus, but further work has to be done as to whether or not a plant would be able to tolerate the infection, because a little leaf punch is pretty small, and a plant itself can overcome these things. So this is just a summary of what our group has found. I manage this large group with the funds. We have since developed an infectious clone, because at the time we were having to do infestation with aphids. So I had to work with the entomologist to be able to infect the plants, to be able to tell whether or not there was some sort of resistance. One of the main things from the Sentinel plot trial was that it seems to be very regional specific, meaning the southern coastal plain is at higher risk than the mid-south or Texas. And we found that the virus can be transmitted by the aphid within 30 seconds of it landing, and that insecticides will not control virus spread. And I also did some grafting. My grad student did some grafting as to, one, the best way to graft cotton, and two, can you use grafting as another way to determine resistance. So, those were probably two big projects that I've had going on. And I kind of wanted to lead into, how to be successful, or things that I feel like have helped me. I teach my class that, I'm a plant breeder. I know how to improve a plant, but I don't know the traits I'm working with. I'm not an expert in insects. I'm not an expert in pathology. I'm not an expert in statistics, Quentin. But that's why having a team is so important, and being able to collaborate to be able to move forward. And so the first thing I will say is, for those of you that can go to a commodity board, remember the growers, remember the farmers. Like, to me, my ultimate goal is to produce a cultivar that is going to go on their land, and they're going to be able to reap the benefits of that. And so I've tried real hard to build relationships with them. And by building relationships with them, I've been able to, I feel, go further than I would have without. So I've been able to attend a United Soybean Board meeting as a guest for our state rep. They don't typically invite researchers. So I got to see firsthand what these growers are thinking and what kind of needs they're looking at. I attended a Southeast Regional Cotton Commission meeting. And I was appointed to the Technical Committee for Emerging Pests for the National Cotton Council. I have been to their farms. I've seen the things they struggle with. I think it's very important to build relationships with the growers. I've also had some other opportunities. I've worked with soybean test weights that's supported by the United Soybean Board, and I've done projects tied in with the Cattlemen's Commission, looking at cottonseed for feed. And, asking questions and pursuing funding that is relevant for their issues and their needs is going to take you a lot further. The second thing I would say is to leverage institutional knowledge and collaborations at other institutions. So when I started, one of the first things I did was I hung out with the staff and the senior faculty members. We had morning coffee on Fridays, and I made sure I was there. I wanted to know where the skeletons were. Where's the equipment located? What is it that traditionally was mine or in another plant breeding program that could be gleaned? And I mentioned I took over the FOV nursery. When I took it over, she was charging $125 per entry for private industry to have a line tested. I was like, wow, that is not enough for us to go out and count plants and determine, the severity of the wilt. So I said, okay. I sent an email out. I said, I'm taking it over, and I'm going to charge $450 an entry. Now, honestly, I threw that number out there I was kind of pessimistic, but I had a company turn around and said, okay, we're going to put 36 lines in. I was floored. I was like, wow, I just have boosted the amount of money that I can use in my program, and I'm not touching my startup. I got equipment from an old textile lab on campus. The experiment station employees told me about where the other lab was. So I found it quite advantageous to listen to the people that were there and know what resources are available. I checked out a book not too long ago from the library, and it's about, I don't know the name right now off the top of my head, but it was about givers and takers. And as I started to read the book, it was very clear. I was like, I'm a giver. Like, I'm gonna give my time, I'm gonna give my energy, and I may not always get something back for it. And what I have found is that has taken me quite far. So like with the combine, I put a proposal together that said, hey, look, this is going to benefit everybody. And when you go to upper administration and you say, hey, this is going to impact all these people's programs, and this is going to push research and elevate the College of Ag, like, they are more inclined to listen to you. I also found that I can't go and complain. If I have a complaint or I don't like something, I got to spend a little time figuring out a solution. So ask and talk about solutions when there's a problem rather than complaining. I also think it's important to earn the respect of your students and staff by actively participating. So I want my staff and my students to know that I'm not going to ask them something to do that I wouldn't do myself. So at least several times a year, I'll go out and help out with 25 bull pigs and other tasks. I've been known to climb in the back of the cotton picker and collect the cotton. The last one I think I have is ask the question. I think a lot of people just are, I don't know if we're afraid to say no, that they'll say no, but what's the worst? I mean, the worst thing they can do is say no. So I was told, hey, like, I couldn't get a new combine, but then I called a couple people and said, hey, what's the best strategy for this? Determine what the limitation is. Like, why are they saying no to you? And then see if you can figure out a way around that. And I call a lot of people. I know some people nowadays like to text, but I like to call. I think I called you, Simon, because I didn't quite understand something. I call a person, I talk to them, I figure out, what their role is, what's going on. And even taking on this job as cotton and soybean breeder, when I took on this role, there was one other person that had been identified as a cotton and soybean breeder, and they worked with, I believe, Monsanto at the time, Dr. Dawn Frazier. And I called her up and I said, hey, how is this? Is this possible? And she goes, look, if you can breed cotton, that's a tetraploid, you can breed soybeans. Soybeans is easy. Cotton is your hard one. And if you've got that handled, and so that gave me a lot of confidence to say, okay, so we're gonna take a stab at this. So really, in closing, it's a balancing act. There are times where I'm flush with grad students. There are times where I'm flush with manuscripts. There are times where I'm flush with grant dollars. And it's really kind of a roller coaster of where you're at. And sometimes at the end of the day, you're going to leave and go home and your brain is a scrambled egg. But you know what? It's going to be there tomorrow. So as you can see in my talk, I talked about the resources that I really had to gather. So I felt like taking the time to gather the resources and build the collaboration was really what was important for me, because I feel like long-term, I'm going to be sustainable moving forward. So I focused this last year on the publications, getting everything off my desk that had happened before CLRDV hit. And so now I'm at the point where I can focus more on the breeding program. I plan to spend more time evaluating populations. And I've got a couple I've got one cultivar that I'm hoping to have released by the end of the year that's got reniform nematode resistance. And so in closing, I don't recommend upper administration, if anybody's in here, to hire somebody on for 2 major row crops. I think that it's a challenge. And with that, I will answer questions.
I actually have a question. Great talk, really good context for that's going to be something to go back to. One of your first slides, you mentioned that there's a complex between fusarium wilt and root knot nematodes.
Yes.
I've not heard of that. Could you elaborate?
Yeah, so typically what you have is you have root knot nematodes where you have fusarium wilt. And when I say fusarium wilt, I'm talking all the races except race 4. That race 4 is quite different. It's out in the west. But technically what happens is the nematodes will feed on the roots and it opens up kind of like sores. And then the fusarium will go in. So they're kind of, it's kind of together. In other words, fusarium is not near as bad if there's not the nematodes present, but when the nematodes are present, it makes it more pronounced and worse.
Great talk. I have a question about, cultivar release from public breeding programs at cotton. Like what path do you see going forward to have companies pick up lines for trade introgression for breeding or for organic production? Like what path do you see for public breeders to release cotton cultivars or lines that will be used in farmers' fields?
Well, that's a great question because cotton does not have a direct path to market. I didn't really go into that, but traded material is probably, there's 99% of the growers grow traded material in cotton. So my response to that is, I think for me, I want to have something better than what's commercially available to where it'd be highly desirable. And so in my elite trial there towards the end, I'm putting it up against commercial varieties. And so I've got some lines that look promising, but I'm really not at that stage yet to where I've had those conversations with private industry. So I do have connections in private industry to where I feel like I can have conversations with them to see, if there's interest there, but I don't plan to approach them unless I've got something good. In terms of reniform nematode resistance, it'll be probably tested out in West Texas in the organic sector to see if it can stand up to what they've got out there.
Thank you for that great talk. You spoke very practically about how you run your lab almost like a business. Do you have any thoughts on how you specifically identify areas that need investment for the most impact for your program when everything needs investment?
So in terms of, you're asking us how to identify priorities in there. So I think part of this comes from working in Australia and being kind of trained on, I need to be the most efficient that I can be in all these areas. And really it's about eliminating bottlenecks, right? So for me, it's been a matter of low-hanging, I guess, what are the easy things to get funding for, and then what's the harder things to get funding for? Like right now, I'm trying to create a much larger delinting area because that right now is a bottleneck. As I go through more advanced lines, I've got more and more and more seed that's got to be delinted. Like when you get to these large quantities, you put them like in a concrete mixer to do it. And so I think, I guess the strategy has been, as opportunities come my way, I've kind of taken advantage of what I could when I could, rather than saying I'm going to do it in this order. I think, I identified a space. I was able to talk the college into, kind of improving it, and they were willing to improve it because it was a unit it was an experiment station 30 minutes away from campus. Nobody was really doing research, as much research as could be there. So they wanted to see that. They wanted that investment. They wanted more people there. They wanted people to actively be on the station. So it was mutually beneficial for them. The combine situation was one of those things that kind of came out of having a GWAS. Trying to harvest a GWAS and then having to harvest it by hand because you're in group 5s and they're starting to shatter, and it's been raining, and the priorities are someone else across campus who 2 weeks ago, you said they were going to harvest them. So that it really comes out of the needs kind of arose. And I don't know if I necessarily have a systematic approach to how I did it other than just what opportunities kind of came my way and when they come. But I've gotten to where I'm at a place now where if I can spend the money to save time, I'm going to do it. Thank y'all.
All right, we are coming up on our next break period. I want to remind everyone that the poster voting will close after at the end of this break at 3:15. So if you have not voted on posters, please do so before then. Posters are all out in the hall here. So let's be back in here ready for the next the industry panel at 3:15. Thank you. Is here, Dr. Natalie Kaiser, who we heard from, Dr. Patrick Cumby right behind me, Jonathan Vogel from BASF, and lastly, Dr. Massimo Irizzo. The goal of this session is really to give graduate students an opportunity to talk to industry folks about what potential careers might look like, what grad school while working in industry might look like, that sort of thing. So this is open for masters, PhD, postdoc, faculty. Feel free to ask questions as well, but remember, this is sort of an educational opportunity for students. We have 45 minutes to ask questions. You guys have 2 mics here, just passing back and forth. And then the audience, Tiago and myself will run mics to you as you have questions. So with that, let's begin. Does anyone have a starting question?
Hi, Dr. Kaiser, you mentioned in your talk about collaborations being like 50% of what you do. So kind of to all of you, what do collaborations look like in your job?
Okay, you go first. For me, that takes a different form depending on which type of teams I'm collaborating with. So if it's collaboration with other breeders, that looks more like, you know, Teams messaging and calls where we're hashing out strategy. If it's with what we call a functional team, you know, the team that's collecting data, that's more often an in-person meeting where we're, you know, brainstorming around a particular challenge. You know, I showed some images of the digital phenotyping work that we're doing. So that could be, you know, a working session where we're all in the field running peppers through the imaging and making a game plan for how we can make enhancements. So it really depends on the team that's in question.
I'll second what she said. I work for the commodity checkoff program for cotton. So in essence, I'm trying to get people across typically universities or USDA labs to do exactly what she just said. So sometimes it's within a university setting, but oftentimes it's across sites. And depending on the nature of the problem, sometimes I'll involve private industries endeavor as well. We fund primarily public sector people, but I'll reach out to the private industry when the need arises. When you're looking for a certain skill set, for instance.
Most of our collaboration will come through the cooperative programs that we work with. So a lot of that's the projects we manage by the cooperatives, but we have anywhere from 8 to 20 different organizations there. Most of that work is discussed and decided in person, and then the in-kind work that we would all participate in is going to be out managed by the cooperatives. Outside of that, we might fund specific research with a faculty member. That's probably it for us.
I think Natalie covered very well what we do on at our company in terms of internal collaboration as well. But externally, we'll work with academic groups or startup companies, largely for either technology evaluation or where we're looking at specific problem we're trying to address internally with solutions best addressed externally, so kind of reach out or do a particular academic group, or if they sometimes we reach out to ourselves, and then set up something kind of more formal that way. So I've been involved with different types of collaborations that way, with startups, with academic groups, also sometimes even company-to-company collaborations if there's areas where different, larger peer companies want to work together, that happens as well. So collaborations, I think, are, in my particular role, are most of my job, either internal or external, working with internal customers or collaborators, or external as well. So there's a lot of collaboration.
Yeah, so for me, it's if I work with, I collaborate a lot on the project that present work on. So the most challenging are establishing collaboration when you want to do a study across a discipline. Those are the most challenging. One, most rewarding also because in education, and for me, because you really build another discipline. I interact a lot with food scientists and I learn a lot from them, but at the same time, you need to understand what language you need to use them with them, what they don't understand that you feel that you understand. And the same way you need to let them know that, not necessarily the way they talk, I understand. I need first to filter. Then those are the challenge sometimes, but very, very good. And then, in general, I think that important thing, collaboration, I really like the last presentation and previous presentation, the importance of collaboration on the work we do. It's important to establish a good relationship, meaning that, you start to go with a collaborator, you stick to it, you deliver, and then you follow up. If there is a plan that you see that you don't follow your name, people don't follow up or not maintain that commitment, it's better to set that up front, because then otherwise there's a risk that collaboration doesn't complete.
Back to ask a question. So I know there's a couple of different first year PhD students here. So my question to you guys would be, what would you recommend that we do kind of throughout the course of our PhD? Is there anything that you would recommend that we don't do?
So to do, I think when you when students look for a supervisor, I think there are ways to try to understand how much time a supervisor really dedicates to a student, knowing how many time does it need, how does follow up on research. The PI, PI should supervise, should guide the student.